Ladies and gentlemen, distinguished guests, dear colleagues, students and friends, it's my great pleasure and honor to welcome you all to the conference on new genomic technologies, hosted here at BOKU University. And I'm particularly delighted to extend a warm welcome to our keynote speaker, Professor Steve Runo. Great to have you here today. A special welcome also to Your Excellency Maurice Makolo, Ambassador of the Republic of Kenya in Austria, and to Dr. Bernhard Bichler, Head of the Division Research, Development and Corporate Services at the Ministry of Agriculture. Your presence highlights the significance of this event, and we are honored to have you with us today. We are equally honored to welcome guests from a wide range of stakeholder groups in the field of agriculture and life sciences. Among them are also esteemed cooperation partners of BOKU University, such as the Ministry of Agriculture, the Ministry of Innovation and Technology, the Austrian Agency for Health and Food Safety, the Austrian Institute of Technology, the Gregor Mendel Institute, Vienna, the Environment Agency Austria, the Chamber of Agriculture and the Austrian Economic Chambers, representatives from breeding and seed producing companies, experts from the food and feed sector companies, farmers who are at the heart of agricultural progress, colleagues and students from BOKU University and other universities, especially from the Hungarian University of Agriculture and Life Science and the University of Vienna. So we are very pleased that around 180 participants signed for watching this event via online live stream. A warm welcome to all of you joining us online. We are delighted to have you with us and hope that you will find today's discussions as exciting and insightful as the participants on site. We also have registrations from at least 34 nations across Europe, Asia, the Americas, and of course, Africa. This global interest emphasizes the importance of the topic. Thank you to everyone who is joining us today in person and online. And now to the topic of our conference. New genomic technologies, particularly gene editing, have become subjects of intense and sometimes controversial discussions. But what exactly are we talking about? In essence, these new technological advancements allow for mutations, that is, permanent, heritable changes in the DNA molecule of living organisms, to occur at specific targeted positions within the genome. This targeted approach is the key difference compared to earlier methods for generating mutations, such as gamma radiation or X-ray treatment, which result in random and untargeted changes. In the simplest case, an edited plant or animal carries a small change in one of its genes, but is otherwise identical to any other natural individual. However, achieving this small change requires biotechnological intervention. And this is where regulatory challenges arise. Should such edited organisms be subjected to the same strict regulations as genetically modified organisms, the so-called GMOs? Or can we agree on a more flexible but less prohibitive regulatory framework? The European Commission has already proposed such a regulatory adjustment in 2023, but it has not yet been adopted. This ongoing debate highlights the complexity and significance of the issue. Why are we discussing this topic today? The answer is clear. New genomic tools, such as gene editing, hold tremendous potential for advancing genomic research and enhancing our understanding of biological systems. Today, we will showcase several examples that highlight these possibilities. Moreover, these technologies offer promising applications for crop and livestock improvement. We are particularly keen to hear from Professor Steve Runo how genomics research can be used to benefit farmers and consumers alike, contributing to sustainable food production and global food security. Of course, we are aware that new genome technologies are not a magic wand that will solve our challenges in the areas of agriculture and food security. There is no such universal solution. However, breeding and genomics, including new genomic technologies, represent vital pieces of a larger puzzle. When used thoughtfully and responsibly, these tools can significantly contribute to creating a more resilient agricultural system and improving food security worldwide. Scientists around the globe, including here in Austria, are committed to sharing scientifically sound information with the public. And in June 2023, leading academic institutions in Austria, including BOKU University, signed an open letter advocating for informed dialogue on these issues. Our efforts include engaging with quality media and participating in public discussions without fear or hesitation. Today's mini-symposium is part of this broader mission. We look forward to the insightful presentations scheduled and warmly invite all of you to actively participate in the discussions. Please do not hesitate to raise your questions during the panel discussion later this afternoon. Your curiosity and critical thinking are what make events like this very valuable. Before I close, I would like to express my heartfelt thanks to a number of people and organizations whose commitments has made this event possible. First of all, Professor Hermann Buerstmayr for his excellent work in organizing this conference. Thank you, Hermann. I think this deserves an applause. Then a big thanks to the dedicated teams from BOKU Event Management, BOKU Public Relations, and BOKU Media for ensuring that the event and our live stream run smoothly. Also an applause, I would think. Thank you. Then a thank you to the Gregor Mendel Society and the Austrian Academy of Sciences, along with the Gregor Mendel Institute of Molecular Plant Biology for their invaluable support. Many thanks. I would particularly like to mention President Heinz Fassmann, who cannot be with us today due to his commitments abroad, but who is a strong supporter of our scientific endeavors. Thank you, Heinz Fassmann. Ladies and gentlemen, thank you all for your attention, your presence, and your engagement. I wish us all a productive, inspiring, and thought-provoking conference, and give the floor to Hermann Buerstmayr.
Yeah, dear Rektor, many thanks for opening this conference and also for honoring our meeting with your presence. We know your schedule is more than full, so it's really a pleasure to have you here, and particularly welcome to everybody here. I'm very overwhelmed, actually, how to say that we have such a big group of people attending our small afternoon meeting today, either on site or online. So as Professor Schulev-Steindl already mentioned, the main aim is to have this event not just as a mere scientific event, but as a science to public translation event, but with a focus on people who have a good knowledge background already in science. So, and I see this is exactly what we have gathered here today. And before I forget it, also a warm thank you to my colleagues from the BOKU Doc School AgriGenomics. I forgot to mention them. Because they have poster presentations in the back, and during the coffee breaks or during the final break, they will be available at their posters, and you can see what our smart PhD students are working on at the moment in terms of animals or plants or microbes, and trying to understand these organisms better for a better future. So I took the privilege to start with the first short presentation myself, because I thought it's maybe a good idea to start with a quick introduction into what is genomic technologies or new genomic technologies, and what are we talking about, just to setting the scene, only for a few minutes. First, I start with a short definition on plant breeding, and this is my favorite one because it says plant breeding is science, art, and scientific and economic activity to genetically improve our crop plants. And this already tells us everything what we speak about. And this definition is from Rex Bernardo, who was a guest professor a few years ago at BOKU, and one of the best teachers in plant breeding whom you can think of. I had the privilege a few years ago to visit an area which is Palestine around Lake Gennesaret. At the moment, unfortunately, a politically very unstable region, but for a biologist, a very interesting region. If you look a little bit closer into this countryside, you see there wild grasses, and these are our wild relatives, wild wheat, wild barley, wild oat, growing there still in nature. When people started to do plant breeding approximately 10,000 years ago, they started with this type of wild grasses. But they were looking for mutations because these wild grasses had a very bad type of allele that makes the ear, the spike, brittle. This means the seeds fall on the ground and they don't stay on the spike. And one of the first mutations that our ancestors looked for was plants where the brittle stays together, the rachis stays together, and they can pick the whole spike and need not pick up the seeds from the soil surface. And this is a mutation. And after 10,000 years of breeding, we still have the same organism with the same set of genes, but different alleles. So we have selected now for mutations that nature gave us, and this is what modern wheat looks like. Genetically, exactly the same number of genes, but in different versions. This means plant and animal breeding likewise uses historical mutations. And now I jump to new genomic technologies. The term new genomic technologies covers technologies that are relatively recent. One says approximately after the year 2000. So it's already 20 years almost. No, try this one. Have a short look into the storage of our genes. The storage material of our genes is the DNA molecule. And the DNA molecule is a super large molecule and a very efficient storage machinery. Because, for instance, wheat, each cell of wheat has 16 million a text of 16 million letters, 16 million nucleotides. And if we would transfer this into books, it is a library of 32,000 books, each one 500 pages, and per page 1,000 digits. And this is in every single wheat cell. Just imagine how efficient DNA is as a storage material. And it's basically a text. But this text is passed on from generation to generation, not completely free of errors. We may see slight changes, and this is what we call mutations. It's almost error-free, but not completely. Try this again. So this is basically what we would look at the DNA molecule. Yeah? It's a simple text. The text consists just of 4 different digits, and the word length is always 3 words. And with this relatively simple language and text, we can write organisms as complicated as us, because we are based on exactly the same principle for our genes. Now, a word about mutations. That mutations can be induced has been known for a long time, more than 100 years. In 1926, Hermann Müller was one of the first to show that mutations can be induced, and mutations are heritable, so they are passed from generation to generation. And this was already worth a Nobel Prize in 1946. So there was already a Nobel Prize quite a while ago. What do random mutations look like? Random mutations, basically, you find randomly, for instance, if you use a chemical mutagen like ethylmethansulfonate, they will target a G and transfer it into a T, a guanine into a thymine, and that's it. And this occurs completely random. You have no control where. But this has been known for many, many years. Now, the gene scissor or CRISPR-Cas9 came around, and we just talked this morning at the right time, because we now already have a good genetic knowledge that we can use this technology efficient. In a nutshell, it is again a mutation tool where you introduce a small change in the DNA molecule, a tiny one, but now you can define the position. You can tell where this mutation should go, and basically it leads to a small DNA break. But each cell has a very efficient repair mechanism, and it repairs this small change, this small cut in the DNA efficiently, but sometimes not perfectly. You can consider this like if you have a wound, you cut yourself, your skin heals again, but you may see a little bit of a scar. And that's exactly what happens in gene editing. The DNA molecule is repaired, but it may look slightly different than before. It has a small scar. And it introduced a mutation. And now the smart thing is that we can address with a DNA molecule of a certain size the precise position where this little cut and this little scar should appear. It's like the mail address of a letter or of an email. It goes, if you send an email, there are possibly 10 billion possible email addresses out there, but it gets to the recipient and nobody else. And exactly that we can do, or how we can understand these gene editing tools. They are very precise in terms of position. And this was worth a Nobel Prize pretty soon after the discovery, already in 2020, for Emmanuel Charpentier and Jennifer Dona, who first described this tool as a possible technological tool. In order to use this tool efficiently, we need to know already a lot. So it does not stand on its own. It's just a tool. But in order to use it, we need to have a good knowledge, a very good knowledge. We need to know where do we want to make the change, so we need to know the target sequence. So there's a lot of earlier research necessary to find out the target regions where we want to make this edit. We would know what type of edit we want, and we also need a technological tool, a biotechnological tool to introduce this editing machinery into a cell. And this is still transformation. That's also needed, otherwise it doesn't work. Once it works, why is it so interesting? On one side, it is a very smart tool for discovery of gene functions. This means if we have a target sequence and we think this is necessary for disease resistance, striga resistance, a quality trait, but we are not sure, then we can modify this position and see what changes in the next generation of the plants. If we have been changing the right gene, we have now allocated a function to a certain gene. Once we know this, we may also use it to really in a breeding context, and we will hear an example of this later today. Once you know, and this needs a discovery phase, you can use it. And we will hear today how this can be implemented. This is just when you type into a search machine, gene technology, and you look for images, and that's what you get. This is the list of, and this is just why we are speaking here today. The public is pretty much frightened by these words, because that's what the search machines give you. But this is all just rubbish. We never use a syringe to modify a tomato plant. I mean, this is just complete nonsense, but that's how it is brought to the public, in my opinion, in a wrong way. What is plant breeding in reality? That's my favorite image of plant breeding and a new genomic technology. It's innovation in rubber boots. And here we also see and I like this, it's from the European Seed Association. It has a number of tools that we need, and genome editing is one of them. But we also need crossing, we need DNA fingerprinting, we need to do a lot of testing and selecting, and we need tissue culture and many more tools to make plant breeding work. Yeah, and with this was my short introduction into the topic, mainly for the newcomers. I hope I could manage to tell you a few things about it. And now we jump directly into our real scientification. And I introduce now Stephen Runo. Yeah, here we are. Stephen Runo, I basically got in contact maybe half a year ago because I was reading your papers, and basically it was Birgit Dalheimer who mentioned this presentation maybe half a year ago, and then I thought, okay, I have to learn more about this topic. And it was kind of a by chance discovery, because normally I'm not working in sorghum. So I would normally not read sorghum publications, but I found this one so beautiful and so interesting that I got in contact, and then we exchanged a few emails, and after a few emails I asked Stephen, would you want to come to BOKU? Stephen Runo is professor for molecular biology at the Kenyatta University in Nairobi, Kenya. He has a bachelor degree from the Kenyatta University and then a master degree in biotechnology from the same university. Then it was also a good luck coincidence, you told me today, that somebody from the Rockefeller Foundation was visiting your university and talking with your supervisors. And then Stephen was offered a sandwich PhD program funded by the Rockefeller Foundation, and Stephen could do part of his PhD at the University of California, Davis, and at the Kenyatta University. Then Stephen undertook a few further trainings and postdoctoral positions, among those the University of Sheffield in the United Kingdom and the University of Virginia in the United States of America, and then returned as a faculty, as an associate professor, back to his home university, the Kenyatta University. And he uses, and he will show us, interesting and modern tools in plant biology, but with a clear link to plant improvement. So it's not just understanding, it's also understanding and using the technologies. And also, Stephen understands some German, because Stephen also got a Humboldt Fellowship and spent several months on a sabbatical at the Humboldt University in Berlin on a fellowship. And now we are very proud to have you here, and the floor is yours.
Thank you, Hermann, for that really nice introduction. Thank you. I'm greatly honored to be here. Thank you for the very warm welcome, Rektor. Thank you for a really good introduction and your understanding of science and your support for science. Thank you very much, Ambassador Mr. Makolo, for honoring us with your presence. I'm really happy and, again, greatly honored to be here. I don't take this for granted. And also to just share with you some of the work that we are doing at Kenyatta University, but also in a broader sense to see how technology can be used to improve livelihood on a very passionate subject that I hold dear and dear to my heart, food and nutritional security. So thank you again for the opportunity. Yeah, I'll be talking about leveraging genome editing, and thank you very much, Hermann, for that really good introduction. I think I may have to skip some of my slides. And I think for me, a good point to start here is to put this conversation and frame it in the context of food security in Africa and farming and agriculture in Africa, because this is different than most regions in the world. Unlike here in Europe and unlike the United States or in South America, for example, farming or agriculture in the context of sub-Saharan Africa or in Africa is very different. This is carried out in smallholder farms called chambers. These are 0.5 acres to maybe 2 or 3 acres maximum. These are heavily infested with pests and diseases, and these form the basis of the livelihood for the family. So the family will depend on this piece of land for their upkeep, so means food, and also to get income from it to buy other supplies for school, for example. So any little pressure on these small farms or on these shambas, any little pressure on this subjects the family or most families into food scarcity and loss of livelihoods. And that's why we are very passionate about food security and trying to use new technologies to improve food production, or at least reduce the level of infestation of pests and diseases. So in my talk, I'll talk about genome editing as one of the technologies that we are using to improve livelihoods in Kenya using striga-resistant material or crop-improved material. And then Hermann did a really good job on differentiating genome editing with other technologies. That was part of my discussion as well, so I'll touch on that. And then I'll go into specific examples on how we can use genome editing to improve food security in Africa with examples that we are doing from our university, together with other examples that have been used by other colleagues. And then I'll end with just giving an overview of what the challenges are, because it's not always a very smooth walk in the park. There are challenges, but there are also opportunities of genome editing in Africa. So what's genome editing and how does it differ from other technologies? So again, this was explained very well, and I'll use this cartoon to just hammer the point home. In classical breeding, you'd have one variety or one crop variety, in this case, maybe the old variety or the wild wheat variety, and then you need to cross those to obtain a new trait, and you'll be passing this on to an elite variety. But you'll need to do many cycles of backcrossing or replacing the old genome that you imported from the other variety, and this takes a long time. This takes 8 to 12 years for you to come up with a variety. You could also use mutational breeding, as was discussed here, and as it is done here in Vienna by the Atomic Energy Agency here. And in this case, you'd get an elite variety, so you'd start with a variety that farmers like, and then you subject it to mutagenesis. Mutation is not a necessarily bad thing, and you're doing this mutation with the hope that it will impact a trait, and then you'd have to select for that trait. Again, this, given the whole genome, the chances of you getting a beneficial mutation using this approach takes a long time, and it's not very easy. So that takes about 8 to 10 years. Or you could use a transgenic technology. In this case, you'd have to obtain a gene from a different organism and then impart it to provide the resistance. Because of the regulations that come with genetic engineering, this takes a really long time, 8 to 12 years. And then there is genetic engineering that was advanced using genome editing. In this case, you precisely identify a gene that you need to alter, and then within the same genome of that organism, then you create very small alterations that are beneficial to provide resistance. And this, because of the fact that it takes a shorter time to regulate, because it is as if it occurred naturally, the mutations are not highly regulated, and this takes about 2 to 5 years for you to achieve a new genotype or a new crop variety. In short, as Hermann said, you need to identify a target gene. So this is your gene editing 101. In 2 slides, you need to identify the source of the gene. You need to identify where you need to make that mutation. And I want to use this example here. Here, this is a very good sorghum variety that has a very good head, very high yields. Usually the case is these varieties of crops that have very good agronomic properties and traits that taste really good to us, tastes also really good to insects and pests. And so those tend to be highly susceptible to diseases. And then this is a sorghum variety here that is highly resistant to diseases, but it's not very pleasant to eat, and it is not very high yielding. The only difference between these two varieties of sorghum is this very short region here. So your first job as a breeder using gene editing technology is to identify that specific region. And once you identify this region, then your work is halfway done. And I want to insist that most of the work in gene editing, or for us at least, starts very, very earlier on. You need to have done your homework and identify the region that is causing adverse effects on your crop. In this case, what regions are causing your plant to be susceptible to diseases. And then one of the best ways, or one of the most efficient ways to do genome editing is using a technology called CRISPR-Cas9. This has a zip code or a short sequence that will guide your editing machinery. The editing machinery here, I'm representing here with this pair of scissors. It's not actual scissors, it's actually molecules that we use to identify regions and target them for editing. And what this does is to just surgically or very precisely remove that region that was causing your plant to be susceptible to diseases. And then the cell will take on its own DNA repair mechanisms, as Hermann explained. And that region is patched, but patched with a new sequence that will replace the old one that was causing disease. And what you have now is a high-yielding sorghum variety that is also disease-resistant. So you've done a very small change on the genetic region that was causing it. Adverse effect, the adverse effect here was susceptibility to diseases. So the only difference is this part. The other plant is the same as the old plant. Or you could do it another way. The part that was causing adverse effects, the one that was bringing about susceptibility, you drop it, and then this is repaired by the DNA repair mechanisms. And what you have is now highly resistant, but also highly yielding disease-resistant variety. So with all this knowledge, with this knowledge of genome editing, is it possible that we can help improve food and nutritional security in sub-Saharan Africa? What can we do with this knowledge of genome editing to help improve nutritional health, food and nutritional security in Africa? And this is a familiar picture to you all because it has been showing since we came in. And I don't want to project the wrong message here. I did not come here to talk about beautiful flowers in Africa, even though we have beautiful flowers that we export to Europe here, but this is not the reason I'm here. This flower here, or these flowers, are really noxious parasitic plants that devastate crop production in Africa. So this, remember the slide that I showed you of a farm, that small shamba, 2 acres or 1 acre. This farmer planted maize. So this is the crop here. But as you can see, the crop has been overtaken by this plant here. This plant is called striga. And it's not a weed. I want to make that distinction. It's not a weed. A weed is a plant defined by biologists or agronomists, a plant that grows where it's not wanted. This is not a weed. This is a parasitic weed. Parasitic weed means that it is depending on the plant for its well-being. So striga is distributed all over sub-Saharan Africa here. There are 3 important species of striga, all of them widely distributed in sub-Saharan Africa. And I say striga is actually an African problem. So if you look here in Europe, you don't have any striga, no striga at all, thank God. And we only have some striga in Southeast Asia, very small infestations of striga, but it's not a problem in Southeast Asia. And in America, there's no striga at all. But if you're very keen, you'll notice a very small dot here. And if you're familiar with the American geography, then you'd recognize this as North Carolina. So a long time ago, there used to be an infestation of striga in North America in the 60s. And this caused a lot of panic. And in fact, the Americans have what they called striga eradication program that spent millions upon millions of dollars to get rid of this small infestation. So took millions of dollars to get rid of that small infestation. Not get rid, but manage it. Imagine how much it would take to get rid of all this infestation. So we know that Striga causes 30 to 100% in yield losses. 30% is when you are very lucky. At the minimum, that means when you plant your crop, you are guaranteed of losing at least 30% of the yield. At the worst and more common, you lose 100% of the yield. It means total crop failure. At least 400,000 of hectares are infested all over Africa. And these are old statistics. I think we should update this. We estimated that to be about $7 billion annually, but these numbers have gone high because we know that Striga keeps expanding in its natural range. So there's more areas that have been infested and more crops are becoming more susceptible. And even the range, the species range has expanded significantly. So why is it a very successful parasite, and how do we come in with genomics to help develop interventions using genome editing? So Striga is really successful. One of the reasons Striga is very successful, and probably this is one of the slides that I want you to pin at the back of your mind because most of my talk will be based on this slide. Striga seeds are really, really small, and they stay in soil for a very, very long time. And they'll not germinate until there's a crop, and they know that there's a crop because when you plant maize, or when you sow sorghum in the soil, it produces biomolecules, which we now know to be plant hormones called strigolactones, which are what I'm representing here. So Striga perceives this and knows that there is a host. Remember, it's a parasite. It is dependent on the host 100%. It cannot germinate unless there is a host. If it germinates and there is no host, then it dies. It's suicide. So it knows that there's a host by these molecules that it produces, that the host produces, called, now we know them as strigolactones or plant hormones. And once Striga perceives these molecules, within a period of less than 6 to 8 hours, then Striga germinates. And when it germinates, it grows towards the host cell. I want to play this video. Play. Yeah, grows very fast and very furious towards the host, and this is guided by chemotaxis. Yeah, grows very, very fast. It digs into the host and establishes connections with the host. This period between germination and between attaching and growing into the host and establishing a connection between itself and the host takes a period of less than 48 hours. So within 2 days, Striga has germinated. The third day, it has already dug into the host and established a connection. And here you have this. This is a transverse section showing Striga coming in and establishing a connection with the host. These pipes here are what it uses to suck out nutrients and to kill the plant. And at this time, it's game over for the host. Remember, this is 3 days after planting. The plant will emerge from soil in 3 or 4 days. And as it emerges, it is already sick and irreversibly retarded in its growth and growth compromised. And we know this is not just an act of stealing nutrients. We know this is also an act of Striga reprogramming the host to make it a good host for itself, to steal food, and also to inject phytotoxins in it. Anyway, at this time, then it is game over for the host, and you cannot recover from that infection. So what we do, we use genome editing and molecular genetics to try and develop interventions. And I'll just give examples today on what we have done on Striga rice interactions, on Striga sorghum interactions, and Striga maize interactions. Remember, this species of Striga hamontica, the most destructive, is parasitic to maize, millet, sorghum, rice, and really all cereals. There's not a cereal that I know that is not infected by Striga. So I'll just very, very briefly go through what we have done so far, how we have done it, and how we hope to move forward. So starting with the striga rice interactions and what genome editing tools we have in place and what we have been doing so far. So I started by saying that what you need to do first is to understand the biology. We understand the biology of striga, but striga interactions with rice. And unfortunately, it is not the same. So it's not one size fits all situation. It's not that a solution in rice would work in maize and a solution in maize would work in sorghum. So every situation is unique in its own interaction between host and parasite. So unfortunately, we have to go through each one of those. And when we started this work, we knew nothing about striga infections and interactions with rice. And we did not know from a perspective of molecular biology what the molecular exchange between rice and striga is to enable us develop interventions for genome editing. So we wanted to know the mechanisms that underpin resistant responses in rice and susceptible responses, and see if we can be able to understand the biology and then use genomics and genome editing to develop interventions. So we wanted to determine the mechanisms of interactions and then use genome editing. And I just want to point out that we started with a rice variety called Nipombare. This is striga, the green part. This is a lab experiment that we do. This striga infections on a susceptible rice variety called IAC. And then this is a really resistant variety of rice. We use nature because we know nature provides solutions. You just need to look long and hard enough for them. There are varieties of rice that are resistant to striga. We need to know why they're resistant, and we need to know how we can transfer and impart that resistance into varieties that farmers like that are also striga susceptible. So this is IAC, which is highly susceptible, and this is Nipombare rice, Japonica rice, which is highly resistant. We scored this using how many striga attached, how long, and what is the biomass of striga. So using this matrix, we are able to define very well a resistant response together with a susceptible response. So what we did was to do an RNA sequencing experiment. This is just a gene profiling experiment that tells you if you have a resistant variety of rice and you have a susceptible variety of rice, and infect both with striga, what is the molecular biology response? In other words, which gene activities are induced in the resistant versus the susceptible variety. And in many experiments we have done in rice with RNA sequencing or profiling of gene expression, we expect to find cell wall degradation enzymes, you expect to find lignin, and we did find this. But what really, really stood out was a gene that had nothing to do with cell wall, a gene that had nothing to do with typical host pathogen interactions. This gene here. So this gene was the most induced. And even though we have a story about other processes that are helping Nipombare cope with striga, this piqued our curiosity. So a closer look at this told us that these are genes that were inherited from wheat a long time ago. And these genes are in a cluster. And not to go into a lot of technical details about this, these genes are responsible for allelopathy. Allelopathy is when a plant grows and then stops other plants around it to grow. So Nipombare has a mechanism that it produces molecules, and these molecules are toxic to striga. So it's able to grow and specifically exclude striga from growing. And we saw that based on all these genes here that we got from this cluster of genes, clustered together in rice and inherited as a cluster when rice was, during the evolution of rice, that formed allelopathic molecules, or what are called phytoalexins. So this is the pathway, and I don't want to be into so much science, but all you need to know is that this molecule here, that is exuded into the rhizosphere is the one that is allelopathic and is the one that is causing striga to die in Nipombare. So our job was to, again, look at this pathway more closely and validate these experiments. So if you take, these are striga seeds, by the way, if you take striga seeds and get the root exudate from Nipombare, striga will not germinate. If you take root exudate from IAC, the susceptible variety, then you have striga seeds germinating. If you use a positive control here, then you see all the seeds have germinated. So what if you take the root exudate from Nipombare and then add it to IAC, that shuts down germination of seeds in IAC. So that tells you that there's something in the root exudate of Nipombare that is shutting down germination of striga seeds. And the same thing, if you take Nipombare root exudate, you put it into GR24 or natural germination stimulant, it shuts down the germination. So that tells you that there's a molecule that is excluding germination in striga. And we think that that is because of the allelopathy based on the gene expression profile and based on the information that we already had. So what we did with genome editing was to test this concept. If this is the path that leads to production of this allelopathic molecule, if you switch off this gene here, then you don't have that activity going on, and all the metabolites are channeled towards here. So we have this cooperation with ISU, where we have edits of rice, susceptible rice, that are being tested for resistance to striga because of production of these allelopathic molecules. Then sorghum. So this story starts a while back. In 2009, Professor Ijeta Gebisa was given the World Food Prize, or was awarded the World Food Prize, because he discovered that there are natural variants of sorghum that are resistant to striga. By the time that Professor Ijeta was getting this World Food Prize in 2009, it was not known what was causing this difference. You have here, this is a sorghum variety that has been infested with striga. This is a sorghum variety, same field, not highly infested. So what is causing this resistance? And it was known much, much later by a group in Purdue that he heads much, much later, this is 2017, that it was because of a mutation in one of the chromosomes of sorghum. And a group that I worked with at Penn State figured out that these mutations is actually very, very conserved in germplasm that are in Africa. In other words, if you pick all sorghum varieties in the world, and then you find the ones that are domesticated in Africa, there is a high bias of these alleles that are resistant to striga. And I mean, that is expected. If host and parasite coexist for a long time, you'd expect that each one of them would be developing strategies to overcome the other one. So sorghum has inbuilt resistance towards striga. So long story short, in 2017, this mutation was identified. And what we did with my group at Kenyatta University, and this is work of a PhD student that just finished 2 years ago, we picked all the sorghum, or most of the sorghum germplasm that have a bias for representation in Africa. So we want to get as many, this is about 314 genotypes of sorghum, and these nicely overlap with the striga natural range. So this is where striga is found in most of sub-Saharan Africa. And sorghum is really, really, really good for genetic studies because sorghum has many sub-races and many races, and each one of them can be crossed towards each other. So there's a high genetic diversity in sorghum represented by this phylogenetic tree. So we have all the races, and these races of sorghum were domesticated at different times in different parts of Africa. So the range of sorghum and striga overlap very nicely, so you'd expect that you'd have resistance very well selected for striga within sub-Saharan Africa. So what Sylvia Mutinda did, my PhD student, was to just screen them for mutations. So screen all those sorghum varieties, use a very simple technique, multiplex PCR. So here is the deletion in the Ejeta work. And what we did was to just find a way of identifying this deletion. And out of the 300 and something, 390 sorghum genotypes, we identified 12, or at least 12 sorghum varieties that had a mutation on the region that was causing striga to germinate. This region is called the LGS1, or the low germination stimulant region. In other words, if you have a deletion, if you don't have this gene, then you don't stimulate striga to germinate. And these 12 genotypes of sorghum were screened in the greenhouse, were screened in the field, and this is one of our field studies here. This is, again, a uniformly infested field, not as dramatic as Professor Ejeta's sorghum that won him the World Food Prize in 2009. But you can still, this is a checkered box. You can see that there is striga infestation here, but no striga infestation here. So the deletion was the one that was causing striga to germinate, to not germinate in the resistant varieties. So we have done further work on this. Yeah, should go. Yeah, further work in this, in testing the concept, if you have that deletion removed using genome editing, are you able to have or imitate that same mutation? So we did that as a proof of concept, and we were able to see that there was reduction in the germination stimulation using genome editing. So in maize then, again, we rely a lot on nature. What occurs in nature will most certainly be sustained. Don't worry about that long biosynthetic pathway. That was just to scare you for showbiz. What I want you to focus on is these two molecules. Again, like in sorghum, there are two forms of germination stimulants. One is effective, the other one is not effective. This was work that was done in Amsterdam, the lab of Hermann Buerstmayr. So they identified the biosynthetic pathway of synthesis of germination stimulants of strigolactones. Appears there is two forms. Zealactol is present in resistant maize. Zealactone is in what maize varieties have. So when this paper came out in 2023, one of my PhD students said, why don't we test all the maize that we have in Kenya, see which zealactone they produce, and that began the process of a new project. And in this project we did, we took several land races of maize and checked for how they stimulate germination. And it turns out that there is a very big difference in the way that these stimulate germination, okay? So they stimulate germination. It could be anything. It could be any strigolactone. It could be other molecules that are in the blend. Then why not check for the levels of mutations? So we check the mutations on the key genes that are involved in the zealactone and the ones that are causing zealactone and zealactol conversion. And indeed, we found mutations. Some of them are stop codons. Some of them were just deletions that changed the way that the gene is read, what we call the open reading frame. And then we measured the levels of zealactone. So are they producing zealactone or are they producing the other molecules? So this is an experiment that was done by our colleagues at KAUST, King Abdallah University of Science and Technology. And some maize varieties were producing zealactol and zealactone, then methyl galactonic acid. But that's good. But what we need to do is to relate the mutations with the compound that is produced with the germination, and then move to genome editing. So we found that these were correlated. The resistant varieties, as predicted, were producing more of zealactone, and then, the susceptible were producing more of the zealactone. The susceptible were producing less. So this interconversion, metabolite interconversion of zeolactone to zeolactol impinges on germination stimulants, and then consequently on the germination stimulation. So these are the two molecules that we're working on, and we're working to change the flux of zeolactone so that they don't produce zeolactone, they produce zeolactone. And this just means that in one of the biosynthetic pathways, you shut down one path. It's like closing one tap and then letting the water flow on the other path. And so far we have started to generate these mutant lines. So I've only talked about maize so far, but maize is not the only food that we eat. This is a slide that I got from a review that we did together with Lina Tripathi, looking at the food security foods in Africa. We eat a lot of bananas, we eat a lot of cassava, we eat a lot of maize, and then what we need to do is to develop interventions for all this. And I'm really happy to report that in addition to the work that we are doing, other colleagues in Africa and in Kenya specifically are working on other crops. This is maize lethal necrosis. This is a disease that devastates maize production. This is a virus disease that works in combination. Two viruses work in combination to cause devastation on maize production. This is called maize lethal necrosis, and there are gene editing interventions already being developed, in fact, really, really advanced. And then these are banana diseases, Lina Tripathi working at IITA, and many other interventions. And this slide, I think, highlights that, and I just want to mention a few. I just got this from one of the reviews that we did, to highlight some of the work that is ongoing in genome editing. There's even animals, development of vaccines for animals, the development of rice, resistance against bacterial diseases. There's work in wheat going on in Egypt. There's work in yams, there's work in sorghum, there's work in bananas. And I know, even though not in Africa, and this is something that I'm looking forward to, there's work going on in the University of Queensland to develop apomixis, and this is fixed hybrids of sorghum as well as cowpeas. So this means that farmers will be able to produce hybrids and then share them the season after season after season. So that means that if you don't have to go to the shop to get a new hybrid seed, you can in fact fix that hybrid vigor for a time as long as 7 years. And I'm really looking forward to that time because I know that is being developed, and I know there's a proof of concept. I know that there's work that is ongoing right now on development of hybrids or apomixis in rice, fixed hybrids in rice as well. So there's a lot of things in the pipeline in Africa, and I think the future can only be brighter in terms of food security if all these projects come to fruition. As I finish, I just want to highlight some of the challenges and opportunities that there are. So I just probably painted a road that says everything is happy-go-lucky, everything is okay. It's not really true. We have challenges, and I use this hourglass to represent the challenges of genome editing, but also represent the opportunities. Some of the challenges we have is research capacity, and I think we should leverage and have partnerships that help build our own research capacity. I think we have started, and I think we need to develop this as much as possible so that we have and sustain this momentum for genome editing, and that's really important. One of the things I'm really keen on and passionate about is developing local capacity. That goes alongside having functional labs, and then resources to conduct research. And as I give my acknowledgement, I'll mention the government of Kenya is also really keen on supporting research, and actually supported some of our work. But then we also have on the demand side, this is like a seedling that is being given nutrients. We have really good regulatory structures in Kenya, particularly that are grounded and based on science and based on end product regulation. So we have the goodwill of the National Regulatory Authority, we have the goodwill of the government, so we can be able to nurture our projects in genome editing and prospects of using genome editing for food security. Yeah, so again, this is just to highlight the regulatory landscape in Africa. We have the countries that here depicted in green, like a traffic light means go. The ones depicted in orange, or the color that women call yellow, here means wait. And then this here, it means that they are regulated as though they are genetically modified. So even though we have these 6 countries now that have genome editing specific guidelines, there are still disparities in regulation of genome editing, and that presents challenges for exchange of genetic material and also for scale-up. So this is my last slide. I hope by now I have convinced you that Africa has potential to increase agricultural productivity using genome editing. I hope that was clear. And I also hope that going forward, what we need to do is to improve our capacity, because this is one of the choking points in my hourglass. We need to leverage partnerships in order to develop our capacity for genome editing. And this will help us develop demand-driven genome-edited products, products that solve and address societal needs. Societal needs, I said, Striga is a problem in Africa. Striga solutions must be found within Africa. And then commercialization of gene-edited products would require proper regulation. So we have regulatory authorities that are bringing in place good and sound and must be grounded in science. And this will create a good enabling environment. And then finally, there are genome-edited products that are advancing towards commercialization in most parts of Africa, and that provides the much needed impetus to develop further products. So I want to thank very, very sincerely by Kenyatta University. Some of the work was actually funded from the Vice Chancellor's grant. That's the reactor here. So seed grants to help us prove some concepts from the university itself. So I really appreciate it's modest, but it moved us forward. And then funds from the National Research Fund. This is the funding body in Kenya. We got most of the work, substantial work in sorghum was funded by the National Research Fund. And then I also got some funding from the Royal Society that came with the prize that I got in 2020 that also set the pace for genome editing work, really started off on genome editing in sorghum. And then I also have and acknowledge funding from the International Center for Genetic Engineering and Biotechnology. I have partnerships with Corteva. I don't know why their logo is not here, and Penn State. And with that, I thank you very much for your attention, and I'm happy to take questions.
So, Steve, thank you very, very much for an interesting and inspiring talk. Since we have a long time for the discussion of the project as such, I would suggest that we have these questions at the end of the sessions. So if there's an urgent question for understanding something that was said, we can do it now. And otherwise, in the interest of time, we would go on and have the long discussion later. Okay. It was crystal clear, so there's no obvious question in that aspect. Well, we have seen this great potential in this very wide context and a lot of active work going on to use the potential. But also here in Vienna, we have quite some attempts to use the potential of the technology. So it's my big pleasure to welcome 4 young scientists, more or less young scientists, to present us these examples of ongoing work here. And we will start with Irma Tkachenko from the BOKU. She's working in the lab of Christian Luschnik, and she will give us an example of how to use gene editing in the context of abiotic stress resistance, right? So Irma is from Russia, but she studied in Spain and is in the final stage of her PhD work now. So very welcome, Irma.
This is the last point. Okay. Thank you very much for very kind introduction. I'm very happy and excited to be here to share some results about my project. So, imagine a world where farms struggle to produce food not because of lack of seeds or poor soil, but because there is simply not enough water. Water scarcity is a global crisis driven by climate change, population growth, and poor exploitation of water bodies that will be affecting all the continents by 2040. Water management and drought-tolerant crops are essential in food security. To tackle this question, my colleagues from Corby Group have been studying a protein family called tolls. They are important in controlling specific protein turnover, meaning they take care of potentially useless and dangerous proteins. My colleagues found out that if you turn off or turn out the tolls number 2, 3, 5, and 6, out of model plant Arabidopsis thaliana, you will receive a mutant which show drought-tolerant phenotype, which can be seen here on this picture, showing the survival rate of adult plants after 3 weeks of drought stress. Here we have a quadruple mutant versus wild-type Arabidopsis and 2 mutants. One of them is highly susceptible to drought, and another one is highly tolerant. So what's most interesting is that this drought tolerance did not affect further phenotype of the plant, and potentially it would be very interesting and valuable trait for crops because altogether the fitness of the plant and the yield won't change. So, as I already said, we would like to transfer this knowledge from model plant Arabidopsis thaliana to the crop, and our crop candidate is soybean, which you all know by tofu, soy milk, and soy sauce. Soybean is important for seed protein oil production, for nitrogen fixation, and also it drives an increase in production of the beans. It has a complex genome, it's paleopolyploid, and it means that the gene of the soybean went through duplication at least twice during the evolution, and that explains why many genes are presented in copies. One of the methods that really pops up is CRISPR-Cas9, which allows precise modifications in DNA. And the popularity of this method can be reflected in this chart showing the number of publications mentioning CRISPR-Cas in plant field. CRISPR-Cas9 is a genome editing tool that allows precise modifications in DNA. It is based on natural immune system of bacteria. And it also has 2 major components. So Cas9 that act as molecular scissors guided by RNA to the target site. So this genome editing tool allows gene modifications, insertions, and corrections. And the following, the general scheme of applying CRISPR-Cas9 goes as follows. First, you would like to identify the targeted genes. This can be done by navigating databases. And then you would commercially obtain these two parts of the CRISPR-Cas. Usually you receive them in the form of a plasmid, which is a circle piece of DNA. But you can't just directly insert it into your plant. You need a mean for that. And one of the options would be Agrobacterium. So Agrobacterium, it's a specific organism used for production of transgenes. In nature, it induces tumors in plant genome by inserting a tumor-inducing tDNA into the host genome. But the researchers have learned how to make Agrobacterium transfer any desirable piece of DNA into the plant without inducing the tumors. So once you have your plasmid inserted in the Agrobacterium, you also would need to have some explants of the plants available for the infection. And after some tissue culture, you will hopefully receive a transgenic plant. In case of soybean, there are 18 orthologs of tolls predicted for Williams 82. It's a cultivar soybean that I'm working with. And overall, we are targeting 10 toll loci. So we want to employ CRISPR-Cas to produce knockouts of these genes and to study the consequences of it. My main task in this project is to establish soybean transformation and regenerational protocol for two constructs. First is CRISPR-Cas, and the second one is Gas reporter gene. So reporter gene is a genetic marker used in molecular biology to study the expression of the gene, the promoter efficiency, and also to assess the transformation efficiency, because it produces a measurable signal such as change of color. And this is the precise reason why we use it in this essay. The whole method is quite time-consuming, but also it's straightforward. So first, you would like to have your seeds sterilized and then soaked in water, and then you would need to prepare the explants. So in this case, you would need to precisely cut the seed into halves, so each half contain the embryo. And at the same time, you also need to have your agrobacterium suspension ready, because the next step is to co-cultivate your agrobacterium with the explants for the bacteria to infect. And then you start with the selection process based on your selection marker, and also by producing the organogenesis, by mixing different phytohormones in various compositions and ratios. And then hopefully, after a quite long process, you will end up with a putatively transgenic plant. So some of the results assessing the soy transformation efficiency include the PCR test for the presence of the transgene and also Gas assay. So Gas is one of the reported genes that can be detected in case blue color is produced during the staining reaction. Such enzymatic activity indicates the expression of the transgene, and it tells you that the transformation process works. Some other results concerning the CRISPR-Cas also include the PCR testing for the presence of the transgene, and also amplification of the targeted tolls, purification of those PCR products, and sending them for sequencing. And this is the usual result that you get. So these peaks stand or represent single nucleotides in wild type on the top and on the bottom in the actual explant. And crowdiness and overlapping nature of these peaks indicate a possible polymorphism going on at this target site, implying a CRISPR-Cas event happening at this target site. So again, telling us that the approach works. Some of my current tasks in this project include the genotyping of the plants and selection of the transgene on selective media. This also goes for the next generation, and the further cultivation of these plants in greenhouse conditions to have a big number of population to be tested via phenotypical analysis. With that, I would like to thank you for your attention. Also would like to thank my team and FGG for funding this project, and also last but not least, all the people and the teams involved in organization of this event. Thank you very much.
Thank you, Irma, especially for staying in time, so we have time for 2 questions. Maybe then I start with one. Transformation is definitely a rate-limiting step in the application of gene editing. So how do you calculate how successful you are in the process? How many mutations you get per experiment?
Well, actually, of course, it's a very it's a complicated process, and the efficiency really depends on the cultivar that you're working with, on the strain of agrobacterium you're working with. So many things affecting this result. So in this particular case, we assess the transformation efficiency by the PCR test and also by the Gas assay. So it really depends on your particular situation, I would say that.
Thank you. More question? And maybe later, during the coffee break. Thank you, Irma.
Thank you very much.
And then we come to the next contribution, which is given by Herbert Michlmayr. Michlmayr. Yeah, that's right. He is an experienced scientist working in the group of Gerhard Adam, also at the BOKU, and he will give us an example how to target a biotic stress for plants in case of resistance, if I know right.
Yeah, so, well, I will come to it. Yeah, so first, thank you for the introduction. I'm not only working with Gerhard Adam, I'm also working with Hermann and Barbara, so from the biotechnologies. In the moment, I am in between two worlds. But yes, so let's get to the point. So I am now talking about wheat, and wheat is quite often infected by fungi, mainly, or among other species, of course, Fusarium fungi. So in wheat, this disease is called Fusarium head blight. Oh, this is sluggish. I think for obvious reasons. And it does not affect only wheat, but the main cereal crops we know. The effects are drastic reduction in yield and quality, and also we have contamination with mycotoxins. So in a ranking of wheat diseases, Fusarium head blight is in the top 2. And I think it's difficult to really put in numbers, but the estimates are that on a global scale, the yield losses due to Fusarium infection are roughly 20% in wheat. And the other problem are mycotoxins. A result is that the main cereal products we consume are regularly contaminated by Fusarium toxins. These are, well, they possess different toxicity, and there are guidelines and limits in the European Union to limit maximum levels, but this doesn't mean we are on the safe side. Actually, if you consume a lot of cereal products, it is quite likely that you exceed the recommended daily intake, and you will have a lifelong exposure to these toxins. We probably all have, unless you do not consume cereal products, which is most people don't. So how can we deal with this problem? There are different traditional agricultural techniques and fungicides and so on to deal with the problem. But we believe that finding natural resistance to Fusarium infection in wheat and other cereals is the best thing to do. So we do not know of a wheat variety that is completely immune to Fusarium or fungal infection, but we know that, I have no idea how many wheat varieties actually exist in the world, but in natural lines we find resistance to different degrees. And this is why I would like to introduce you to FHB1. So in the early 2000s, it was discovered that a particular wheat variety has pretty good resistance to infection. And until now, this is still the most effective Fusarium resistance factors we know of in wheat. So it was surmised that there is a hypothetical resistance gene that was called FHB1. And until now, we don't exactly know how resistance works. And it is also quite difficult to isolate the resistance gene, considering, Hermann has mentioned it before, wheat has really a quite huge and complex genome, and it is quite a difficult task to isolate one single gene that causes this resistance. So the main way to identify such a gene is resistance breeding. So assume you have a wheat variety that contains or has desired properties like high yield, and you want to make it resistant, you will cross it with a resistant variety that may have less good agricultural properties. You get a mixed population, you select resistant lines, back-cross them with your desired variety. You do this several times, and in the end, if you're lucky, you get a wheat line that has this resistance. And this means also that when you compare the two lines, you have two lines that are almost genetically identical except for the one resistance gene. And this is why when we conduct genotyping, genome analysis, we should be able to find this resistance gene. Of course, that's not quite easy. It's a lot of work. And so it was first discovered that this resistance gene FHB1 lies on chromosome 3b. But it is quite, the more you want to get into detail, the more difficult it gets. And this was work done mainly by Barbara and Hermann in the last years. So we had a fusarium project called the SFB fusarium, where they spent a lot of work to map this gene, or it's not actually a gene, it's a genomic region, and they could identify a region containing, well, roughly 28 genes, and it was clear resistance must be encoded somewhere here. But, well, we have the basic biochemical knowledge, and when we look at the function of these putative genes, it's not obvious which one would be the actual resistance gene. So the idea to clarify is to narrow this region down. And there is also at the moment a FWF project going on, coordinated by Barbara. And the idea is to now really elucidate the actual mechanism behind FHB1. So one part of the project was to narrow down this region. This was still done by more classical approaches. And at the moment, it's a little bit simplified, but at the moment we have a relative small region that contains a few genes that are potentially interesting. And of course, we look at this in different ways. We can form hypotheses and we test them in different ways, but CRISPR is in principle one way to really test single genes. So we can, let's say for example, we could delete this gene, and when we lose fusarium resistance, then we would be pretty sure that this gene is the causal factor, and so on. Of course, it's not that simple, but so this is what we're working on at the moment. So the gene editing is done by the group of Eva Stöger and Esther. So we grow these edited genes in the greenhouse, and then they are tested with fusarium infection. And this way we would hope that in near future we can understand how FHB1 actually works. So the main points I wanted to deliver is that we believe it's quite important to understand how this mechanism actually works. How can we achieve fusarium resistance? And this CRISPR technology, first, of course, it's not simply done. It's sometimes when we present it, it sounds like it's a tool and you can do it overnight. That's not the case. It's still hard work, but it is a quite important diagnostic tool to identify mechanisms that would take much longer if we do it in traditional ways. Yeah, so to acknowledge, we are grateful to the FWF for funding this work, and this project is also in tight cooperation with the group of Eva Stöger. I mentioned it before, and they do the CRISPR work. So, thank you.
Thank you, Herbert, again, also for staying in time. So, questions to Herbert? Is there any natural variation in that region which you can use from other wheat? Is this Chinese variety the only one that has this natural resistance?
There are, the FHB1 region originated from this Chinese variety, but meanwhile it was crossed into a lot of different wheat lines. But there are other resistance genes, like I think the last estimate 200 reported different similar resistance genes, but in the most cases it's not really validated that provide a high level of resistance. But in the most cases, similar, we don't know how they really work. So there are, actually, I'm not sure if there is a single case where there is an unambiguous solution. I don't think so.
But you're focused on protein coding genes. Could it be that there's a non-coding gene in between?
We do focus on protein coding genes, but not entirely. So it's, well, it's a long story. The truth is we are not the only ones working in this. And in the last years, there were different people publishing, we have found it. And 2 years later, another paper came out, okay, we have found that that's not it. We have found another one, and it's quite complex. So, but yes, it could not be a protein coding gene. At the moment, it looks like there is a protein that is probably really responsible, but still we don't know exactly how. And it's in dispute.
So it's a very interesting, but still a little bit mystic.
It is quite mystic, but it's also complex. It also shows that we do not understand resistance well enough, as we probably thought we do. But it could be that it is probably a small snip in a region that nobody expected.
Then good luck for the future work.
Thank you.
We hope to learn more progress in the future. But it's a great example how CRISPR as a tool can help to identify genes. And I think we will have another example from our next talk. I welcome Claudia Rosa Espinet. She is from Spain. She studied in the University of Lleida and is now a master student at the BOKU. And well, I'm very happy to see also the very young scientist being courageous enough to give us an insight into her ongoing work. So welcome, Claudia.
Thank you. Okay, good afternoon. I am Claudia Rosa. I am a master student at BOKU, and I will present a brief summary of my research conducted in the work of my master thesis, which consists in inducing mutations in a gene of interest in barley plants using CRISPR-Cas technology. So the gene of choice was the FLAP4 gene, which encodes for the fasciclin-like arabinogalactan protein 4. This protein, which contains fasciclin domains that gave the protein its name, has been studied mainly in Arabidopsis thaliana, and different researchers have reported that this FLAP4 protein have a role in maintaining the pectin structure within the cell wall. Also, they have reported that it's required for a normal root growth in salty environments, mainly by generating a loss-of-function mutant. They discovered that root growth was inhibited in this mutant in Arabidopsis in medium-containing salt. And moreover, the BOKU researcher Dr. Seifert also reported that this FLAP4 protein from Arabidopsis is related or has some interactions with abscisic acid and other plant hormones that are involved in the plant stress response under different conditions. And actually, here, I don't know. So in the picture, extracted from a review from Dr. Seifert, there's this scheme representing the hypothetical role of the FLAP4 protein in Arabidopsis, and you can see that it's interacting with a pectin in the cell wall, which is found in the pectin here, and this FLAP4 protein, these structures representing the fasciclin domains, and it also has been reported that it's found mainly located in the cell membrane, and also here having with its COP receptors, having a kind of interaction with the plant hormone precursors. But this question mark here indicates that although it has been reported to have been involved in different processes, their exact function has not yet been discovered. So despite having all this research going on with Arabidopsis thaliana FLAP4, few FLAP studies have been done in crop plants, and more specifically, and to my knowledge, there has been no publications on barley FLAS, although it has been predicted that within the barley genome, there are some genes encoding for putative FLAP proteins. So basically, the aim of my work is to kind of a bit uncover the function of this FLA4 gene in barley, which actually, according to the bioinformatic predictions, it's found two loci, one in chromosome 1H and the other in chromosome 3H. And my aim is to characterize these induced mutations by the CRISPR-Cas system and characterize these mutations in the primary regenerants and also assess in first-generation progeny the mutations, possible mutations, to check whether these mutations are heritable or not. So in order to do so, here like a scheme of both loci, which these predicted sequences encode for putative proteins. And in purple, you can see the theoretic fast domains. And our strategy was to target these fast domains, as in Arabidopsis it was reported that the fast domain integrity was essential for protein functionality. And in order to we will, in these red arrows, I show you that we flank this fast domain to ensure that we will kind of inactivate this protein. So the flanking regions that are targeting the fast domain are the guides that we designed for these experiments and these transformations. And the particularity of these guide RNAs that are, again, flanking the fast domains for one of the loci, have the particularity that they contain a specific restriction sites that were further used for restriction enzyme analysis to quickly check if we contain the mutations in these plants or not, meaning that if there was any mutation going on in this sequence, the restriction enzyme site would be banished, and therefore the restriction enzyme could not perform any digestion, and this could be visible in an electrophoresis gel when compared with a mutant. Also, we included in this vector design, of course, the gene encoding for the Cas9 protein, and also an hygromycin phosphotransferase gene that it was used as selection marker, meaning that only the embryos that incorporated this vector would be able to grow in a medium containing hygromycin. So once we got our vector design, we introduced this vector in immature barley embryos using a particle bombardment system, so a biolistics approach. And we regenerated Kali in selection medium containing hygromycin to generate fully grown plants, which were their genomic DNA was extracted from leaf tissue and further screened for the presence of Cas9 gene by PCR. Once we confirmed presence by Cas9 gene in the genomic DNA, we amplified these sequences from these FLA4 genes, and we conducted this restriction enzyme assay here. Actually, as I mentioned before, to quickly detect whether mutation events were going on in these samples, and once this mutation were confirmed, we send it to sequencing and align it with the reference sequence to check, to further characterize these mutations. Once we had confirmed mutated plants, we put some of these seeds to generate a third generation progeny. And again, once we have fully grown plants from this progeny, we basically conducted these same steps. So as a result, we were able to generate 2 lines containing editing in one or both loci. So for line 1, we got 2 regenerants that indeed got editing events in both of the loci, but for line 2, but for individuals that got editing even in one of locus. So while for the black lettered mutation types only could translate or mean that we only had like a few amino acid loss in the putative protein. For the ones that are marked in red, we could expect a frame shift mutation, which would be quite relevant because it would mean that after this mutation, there would be a change in the amino acid sequence and therefore probably an appearance of a premature stop codon. So here I show a bit that kind of frame shift we have in line 1 in the first locus, and this 23 nucleotide deletion would then mean that there is a shift in the sequence, and therefore like an amino acid sequence changed, and it is not shown here, but there's an appearance of a premature stop codon 33 amino acids before the original stop codon in the putative sequence. Well, as for the frame shift in the line 2, these 7 nucleotide deletions would also cause a change in the amino acid sequence, and in fact here in red, within the red circle, you can see that there's even an earlier stop codon. As regarding the progeny, out from 49 T1 seeds that we germinated from these mutant plants, 7 of them were found to have inherited these mutations, and therefore we could confirm that these mutations caused by CRISPR-Cas were indeed passed to the next generation. So I would like to conclude that indeed CRISPR-Cas system had allowed us to do a precise targeting on the genome sequence, that this kind of targeting allowed to generate frameshift mutations that were really relevant to cause probably to be more likely to have a gene knockout or a loss of function mutant. Actually, my next steps could be to perform an expression analysis to quantitatively assess whether this gene is still expressing or not, and also maybe it would be interesting to do some comparisons with wild-type plants and do some growth assays under different stress conditions. And moreover, I think that this CRISPR-Cas generated, in case that it is confirmed, loss of function mutants can provide new knowledge on the FLAP4 proteins, specifically in barley, and that I think that knowledge on these proteins that are believed to be involved in plant stress regulation can be very useful for, in the future, generate resistant crop plants. Thank you.
Thank you, Claudia. And in the interest of time, I hope you answer questions during the coffee break. So let's go to the last of the short talks. I welcome Mattia Dona. He's from the Gregor Mendel Institute, and he will give us an example how you can use the mutational capacity of CRISPR-Cas for a gain of function and as a tool for studying developmental biology. Well done.
This is for the slide, and this is the pointer. Oh, yeah. Okay. Okay. Thank you very much to the organizer. Thank you for the introduction, and thank you for the previous speakers for having beautifully introduced how to make loss of function mutations with the CRISPR-Cas9. I'm gonna talk about something a bit out of the box of use of the CRISPR-Cas9, and especially in developmental biology. So the question of lineage differentiation, what is a cell lineage? We can say that's the history of in space and time of the division and differentiation events that brings like a pluripotent stem cells into its differentiated, highly functionalized final state. This is, of course, is critical for multicellular and complex organism that is composed by many different cell types. And these in animals has a very strict and rigid determination fate. And thus we know, for this we know in C. elegans, an example, we could map every single cells in the adult with a lineage mapping. And among the tissues in an organism, the germline, so the germline lineage is of particular interest because it's the one who's giving rise to the next generation. What about plants? Yep. Plants have a much more flexible lineage development, and this is due to the plant lifestyle. Imagine a tree that every season has to reproduce all the leaf or the aerial tissues, and especially the germline in plants is very important because it's the one who gives rise to seeds and fruits, which is what we use as food. So how to study lineage development. Well, this is an example, old example. I don't know how to use this. Okay, where random genetic changes leads to activation of transposon that mark cells in red. So there's development of these sectors. These sectors basically represent a footprint of how a single cell developed into its differentiated progeny. But of course, we would like to have something more accurate that allows single cell tracking that is compatible also with the modern high throughput technologies for sequencing and so on. So we thought to use the mutagenic potential of the CRISPR-Cas9, which has been already extensively discussed, not to disrupt a gene and to make a loss of function, but rather to make a gain of function, so reactivate a non-functional gene. What is this non-functional gene? That is our reporter. That is the reporter who will mark the cell that comes after. So I will guide you through how we built these quickly and the modules. So we choose a protein that is highly abundant, very stable, and link it to a fluorescent marker. So this will work as the red that you see before, but just under the microscope and fluorescent. That is beautiful fluorescent green. We introduce a CRISPR-Cas9 target that is synthetic. And the most important part, this gene contains a frame shift. So this protein cannot be functionally translated in normal conditions. So there's no signal in the cells unless we introduce also the Cas9. And stochastically, the Cas9 will produce mutation at the CRISPR target, which might restore the correct sequence, the correct reading frame of this reporter, thus showing a beautiful green fluorescence in the nucleus, which is stable and it can be passed throughout cell division and is stable also in differentiation. So this module is our lineage tracing reporter. That, of course, goes in a bigger vector that we transformed into the plants that contains also the other components of the CRISPR system, which are flexible and can be expressed very specific in very specific tissues, depending on the promoter sequence that we introduce. So we built this in the lab, and we decided to test it first using a very well-known lineage in the root. So the root epidermis development is very well known. So we drive in the expression of the Cas9 in the tip of the root in the stem cell area. And what we can observe is that some of the cells gains fluorescence. The fluorescence is kept throughout the cell division and labeled a cell line, which is absolutely confirmed by the previous studies that this belongs to the epidermis cells that we wanted to track. So this consider it as a positive control of our system that is targeting the right cells, and it shows everything that we wanted to see. But of course, we are more interested in another part of the plants, namely the shoot apical meristem. The shoot apical meristem is the area structure of the plants that contain the stem cells, which give rise to everything that you see above ground. There's still a lot of unknown questions regarding the biology of the shoot apical meristem. We know that it is defined by the presence of certain genes that define the stemness of these cells. And we also can divide this structure into 3 layers, layer 1, layer 2, and layer 3, different shade of green. And these layers are suspected to have a functional differentiation and giving rise to different structure. But again, there's still a lot of open question on how they contribute to the production of all the tissues in the plants. So we applied our system to targeting expressing the CRISPR-Cas9 only in these red cells, as you can see here. And thus, further, we saw that there's stochastic event reactivating one of the cells in the meristem, which later divides and differentiate and give rise to, in this case, is part of the leaf. So we could actually follow back from the differentiated cell track it down to the single cell event that gave rise to that tissue. Yes, then we also evolved our system, I'll leave it there, by make it inducible, which is make it more fine-tuning and applicable for other purposes, but I won't spend time on this. But just by proving and assessing how it works in the meristem, we could actually observe a very interesting thing, that is most of the labeling events are actually occurring only in the layer 1 and the layer 3 of the meristem, and very few in the layer 2. Of course, these would need extensive investigation, which was not the purpose of this work. But there's already opening the question if there's a functional differentiation between the 3 layers. And is it the germline connected to this? That's, I think this is just a beautiful tool that would help researchers in understanding this. As I shown before, this is also stable, so we can do this throughout the life cycle of the plants by following sectors forming in all the tissues and tracking down the lineage development of the single of the different tissues and cells. And of course, we also tried to do it in another model plants because to make it flexible. This is Mercantia. Over time, we could follow the growth of certain specific areas, tracking it down to a single event in the meristem. So this is a bunch of conclusions, but I think the take-home message is that it's very important always to approach things by different angles. So we use the mutagenic potential not for knocking out genes, but to gain a function and apply it to fundamental developmental questions that have also other downstream effect. And of course, we also built this module to be fully flexible and allowing for improving and optimization, which means more colors, different promoters, and this is more details, molecular details, but it can be applied basically to any organism where you can put CRISPR-Cas9. And yeah, with this, I would like to thank you for your attention, and thank you, Austrian Mittelstand Group.
Thank you, Mattia. So we are a little bit over time, and I really don't want to cut the coffee break short. So thank you all the speakers for very interesting contributions. And use the time for communication. Have a look at the posters. And the coffee break is sponsored by the Austrian Academy of Sciences, who also welcomes you for this event on their behalf. Thank you very much.
And I think we can start. As promised, after a more scientific part and scientific discussions in the first round with our keynote speaker, including our BOKU and GMI colleagues, to try to show you that this is not a miracle technology nowadays anymore, that these technologies can be implemented readily by master students. And it works. But now you all know that the political discussion is ongoing, how to regulate, how to treat new genomic technologies in a regulatory framework. And this is, in my opinion, sometimes even more difficult to answer question, particularly in countries like Germany and Austria. But in order to give us some idea on possible implications on agriculture and the agriculture sector internationally and nationally, I'm very happy that Adolf Marksteiner from Landwirtschaftskammer Austria, Federal Chamber of Agriculture in Austria, will explain to us some of these possible and planned and anticipated implications depending on regulations that we can expect. Please, Adi.
Good afternoon, ladies and gentlemen, dear professors. The Austrian Chamber of Agriculture is representing between, I'd say, around 150,000 farmers in Austria. Farming still has an economic and also political importance in this country. I'm head of division of market policies, so my colleagues and me, we cover all sectors of production, from plant production towards animal production, and also the question of organic production, GMO-free production, but also seed production is very important for us. When I had a look before I came here at the curriculum of Gregor Mendel, I realized he was a priest, he was a scientist in mid-18th, 19th century, and those who learned about Gregor Mendel knew what he was doing in 19th century. When you have a look at Wikipedia, you will find that Gregor Mendel wanted to come to Vienna as a teacher 2 times, and he was rejected 2 times to go to Vienna from people who thought he was wrong with his thesis on breeding. No one remembers the people who were responsible for his rejection to go here, but his name survived in science and science of breeding. I want to give you an overview about the sector, and also it will consist more with an analysis and arise a dozen of questions, perhaps not with answers, but it's necessary to give an analysis to start a good and open dialogue. Austria's agriculture, it's marked here by the red circle. Austria is here. Kenya is here. Austria is half arable land, half grassland and alpine land, and almost more than 1/3 of the surface is forest. What I wanted to emphasize is Austria population is still more than 1 promille of global population. So to put it in a right perspective, we are 1 promille of global population. In my analysis, the question also is, will we be right in discussion on GMOs and NGTs, or could it also be the other way around? We are not a majority. We should be open in a future discussion on that. You will find Austria here at the very right-hand side bottom. This is an EU-wide survey, Eurobarometer, from May 21. Opinions on biotechnology and GMOs. And the question the European Commission put to 27,000 people in the European Union, in all 27 member states, the question was, will biotechnology and genetic engineering have positive, negative, or no impact due to your opinion? What is embarrassing is that around 1,000 Austrians in this poll that Austrians are among those who are the least thinking that genetic engineering can have positive impacts. It's even Portugal. You see all these members of European Union, they all are more confident in biotechnology. This is very embarrassing, and this is part of my analysis. Why is it like that? I can imagine perhaps Kenya must be somewhere here. We have still a very vital plant breeding sector in Austria. Successful activities. You see here those breeders who are affected by any changes in European seed law. They are small, they are mostly small and medium enterprises breeding. And you also will see what they are breeding, from soybeans to wheat, potatoes, and so on. Just for information, some of them are also among the auditorium here. What are at the current the basic positions and perspectives on GMO and NGTs? From my point of view, the national position towards GMOs, GM breeding, is dating back in mid-1990s. This is very important, and it's very constant up to now. Economies and societies in European Union, and I'd say also in global comparison, it's open for scientific and economic developments. It's open for diversity, societal diversity, and biodiversity, but not everywhere. The anti-GMO referendum in April '97 brought almost 1.3 million votes against genetic engineering. And against GM techniques. And what was the reasons in that time, I'd say in mid-1990s? Threats of unknown techniques at that time, overall anxiety following Austrian accession to European Union in '95, about security of food, maintenance of agriculture in less favored areas and small structures, and also anxiety and fears against unintended effects, also combined with the widespread use of glyphosate and Bt genes. I remember this because these were the main critics still last week when we had a big Zoom meeting in the German bloc, say Switzerland, Germany, Austria, with the anti-GMO scene. They said our feed and food will not anymore be secure if GMOs and NGTs come into our life cycles. What has been the characteristics for Austria after '97, after this referendum? GMO-free production and organic production were somehow a counter-strategy to industrialized agriculture. Milk, cereals, fruit and vegetables, they turned out to be helpful USPs. What is a USP? It is a unique selling position on the market. For those who are not so in daily contact with market laws, If you go to a big food retailer and offer him a liter of milk, and a German competitor comes, or a Netherlands competitor, it's better to have this comparison. Netherlands competitor comes, I say, you also have a liter of milk. Every liter is offered with €1, and you can say, my liter is GMO-free at the same price. It's a nice to have argument to sell this. So USP, you have to remember that. What was one of the further motives to be against GMOs and NGTs? Avoid dependency or subjection to multinational enterprises who make money by fees. So that's the point where Austrian agriculture and also NGOs turn to say we are against patents in seed production. The multifunctional aspects of agriculture, think of agriculture in less favored areas, in mountain areas in Austria, helped to create and secure national agri-policy schemes and a direct payment scheme, which is still very important for Austrian agriculture. By the way, 26% of Austrian agricultural land is run organic. GMO-free labels, I told about the example of 1 liter of milk. In food, our staples, they are around 2 to 20% of food brands in food retailers. GMO-free production and organic farming and parts of society still have widespread concerns on the trustworthiness of GMO and NGTs. And there hasn't been any differentiation between old GMOs and new NGTs and CRISPR-Cas till nowadays. Although genetic engineering has become very important, say for instance in pharmacology, in medicine and industrial sector. So I have a few questions that could arise, and they're basics for discussion. One of these questions is, do we lose our USPs by using GMO and NGTs? The photo from the land maps says, tells a region, this is Czech Republic, and this is Austria. You will see a difference in structure of doing agriculture. Normally this farmer, under only economic perspectives of competitiveness, would have to leave his land. Yeah? So in daily discussion, political discussion, all this is mixed together now. Can GMO, NGTs threaten our USPs? Next question, do we lose control? This graph was done in the years of COVID-19. It shows the complex production chains of cereals, soft wheat, durum wheat, corn, rye, barley, triticale, oats. It shows where the raw material comes from and goes to animal or human or industrial consumption. And you can imagine if you have hundreds or thousands of players in the whole production chain, this is quite a complex thing. And if you try to trace here from the beginning, say GMO-free, to here to the end till the consumer, GMO-free, it's a very complex situation. This is one of the big questions of those who are against GMO and genetic engineering in Austria. The same picture exists for pig meat, for instance. The NGO discussion last week was around does quality of food suffer? Quality of food will be threatened if new products enter the feed and food chain. Still, 28 years after the referendum in '97. So this is the central slide of my presentation. Do we need new narratives for future agriculture? Say in Austria or in Europe, it's all the same, because the difference between organic production in Germany or in Sweden or in Austria isn't so big, I'd say. Do we need new narratives? It was Urs Nigli who invented this keyword, new narratives. There is admittedly a lack of detection methods of NGTs in a global context. So scientists say that third country products, the detection results will not go more than 40% of all NGTs who are already entering the market. We do not know just what happens on markets. This is not a sign of mistrust from my side. It should be just accepted as a fact. Does GMO-free production have to be redefined? Certainly in food and feed, the mix of GMO, NGT, and GMO-free production, if we would have to trying to keep them still separate for future, it would create additional costs of controls, but probably no extra money on markets. It would just be a nice to have, a USP, or not. We will see in future, but what I think is personally, the big difference between NGTs and old GMOs depending on glyphosate and Bt gene. There is a substantial difference that has to be explained, and I think that's one of the big tasks of science, also to explain what the big difference is. It won't be a farmer who would see as his own task. What we can do is help to support. In case of non-labeling, this has to be changed. In case of labeling non-GMO, non-NGTs, this can cause an exponential rise of costs to maintain a present freedom standard. Please also think of organic. Organic agriculture in Europe till now says we do not want NGTs. And that's a very crucial part of the whole discussion. Does also organic sector have to find a new narrative? Say, this would mean a new narrative with entities, farming with entities. Are there enough advantages you can explain to an organic farming sector? One of the next differences could be the unknown introduction of NGT products on the market could bring a new question. It means we go from soft IP, integrity preserve, to hard IP. So traceability, it's a question of traceability that is very costly. Is organic production and GMO NGTs combinable in its multifunctionality? I would say yes, but the question still is discussed and discussion is ongoing. Is coexistence in plant breeding and patents a future strategy? It is a discussion. Can development and proliferation of successful methods and products, in especially NGTs, be stopped by regulations in open world market? I'd say no. And that's why I appreciated the presentation from Mr. Runo so much, because it gives clear answers to a range of questions. From my point of view, national opt-out is not an option. It's perhaps a short-time illusion. We can discuss about that. And I'm convinced there is a lack of constructive, open, future-oriented dialogue and about the possible advantages. So thank you for your attention. I hope we can discuss about that.
This viewpoint from the Chamber of Agriculture in Austria. Oh, sorry. Yeah, thank you for this presentation and sharing the viewpoint from the Chamber of Agriculture point of view. Are there urgent questions directly to Adi Marksteiner? If not, we can save the questions for the panel discussion. And I would be pleased to welcome Mrs. Birgit Dalheimer. She is from the Austrian radio Ö1 and in the science communication team or science communication of ORF. And yeah, we talked a few times in the past. And so I thought maybe I approach Birgit Dalheimer again if she has time to join this event with us. And also Mrs. Dalheimer has a BOKU relation because she did study at the University of Vienna but did her master work at BOKU. And yeah, and she was working, we were chatting about it some years ago, at the peak time of the GMO debate, and she was working with Birgit Dalheimer, whom many of us know. So thank you very much, Birgit, for coming, and we're looking forward to your
Thank you, Hermann Buerstmayr. I saw some of you laughing when he mentioned the name Birgit Dalheimer. I wonder why. Maybe we can discuss that later. Well, thank you very much for the invitation to speak here. I have a somewhat different angle, obviously, from everything that you've heard so far. Hermann Buerstmayr, you put a title on my talk saying, how can science communication work? I'd say, good question. We ask ourselves that at least once a week. And by we, I mean, do you also hear this sound? Shall I move over there? Might that help? Move away from that mic? Okay. By we, when I say we discuss that very often, I mean a team of very experienced science journalists at the radio station that I work for. Most of us have been working there for at least 15 years, many like myself for many more years. Just as an introduction, for the past year, I've been responsible for a weekly science magazine. This is a 25 minutes program every Thursday night called Dimensionen am Donnerstag, where we try to cover anything from more or less recent publications to current discussions in science. And I would like to share some insights of our daily business, as well as ever recurring, for the past 25 years, I might say, ever recurring discussions on how we do our work, shortly in this talk. But before getting there, just a few words on this science communication and the working of science communication. I guess you will get very different answers depending on whom you ask, obviously. I'll do this very roughly. If you consider science communication as a tool to increase acceptance for science in society, you'll probably address science PR. I don't see it my job to my or our journalist's job to increase the acceptance for science, which is one way science communication is often understood. Then there's a big field that more goes into the real communication, back and forth communication of science. That is everything that has to do with public understanding of science, or the scientific understanding of the public, or even more recently, all that comes along with citizen science. That's again a very, very different area of science communication. And then there's science journalism. Although in some cases the borders might be a little bit blurred, but for obvious reasons I focus on what I think is important for science journalism, for science communication in science journalism. Therefore, let me start by asking whom of you is somewhat regularly listening to radio. Oh, great. Out of those who listen to radio every now and then, who listens to Ö1? Okay, you are a very biased sample, I have to say. We just saw some results from the Eurobarometer in 21. There's a new such survey that just came out last week, and I really didn't have time to go into all the numbers. It's 250 pages long, but I looked at the beginning. I saw, according to the European survey, 17% of the European population use radio and or podcasts as a regular source of information on scientific issues. 17%. For 61, it's TV, still TV, one might say, followed by social media and blogs for 31%. Newspapers range at 19% on the European average, so just a little bit more than radio. And that's all European numbers. It's slightly different in Austria, but not so much, really. So even if it's not the biggest source of information, I'll still mainly talk about radio, obviously, but most of the questions and challenges that we face are probably the same for most of my colleague journalists in newspapers, TV, and so on. I became a science journalist so many years ago, mainly by chance. As Hermann Buerstmayr said, I studied molecular genetics at the University of Vienna, did my master thesis at the BOKU, and then I was about a year into my PhD when the funding of my project ran out, so I had to find a way to earn a living. And applied at Ö1 for an internship. And I had always listened to radio, I had always been fascinated by science, and sharing that fascination and telling scientific stories seemed a worthwhile occupation until the funding might come back. Now you see me, a quarter of a century later, I'm still at Ö1, I have not gone back to university. So that is probably an explanation for my personal answer to one of the first questions that I encountered or not one of the first, but one of the questions that I encountered in my job that come up regularly every year or 2 years or so. That is that general discussion of, is it more important for a science journalist to have a background in science or a background in journalism? There's really many arguments to discuss that. I agree much more with the arguments for the scientific background side, not only because that has been my way, but also it should be self-evident for a science journalist to at least understand a little what he or she is talking about, of course. But I do concede that you can get there by reading and researching. You don't necessarily need a science degree, although I'd say it definitely helps. But what I also think really important is understanding how science works, and that really has accompanied me a lot in my job wherever I go or whatever I talk about. Knowing how science works, knowing how scientists think is something that you sort of insert in your genes if you have worked on science in a while. And having said that, I still have to say that in our really huge editorial staff at the ORF, editorial staff for science, those with a background in natural sciences are still a really small minority. So how do we work? I'd like to give you a really very, very short glimpse of our daily routines, as well as the thoughts behind it. And I'd then actually be happy to hear what you are really interested in. Maybe one of the first questions that I always get asked is, where do you get the ideas of the stories you tell? And who decides what stories come on air, in the paper, wherever? The answer to that question really largely depends on the programs, the format, and the media you work for. As I was sitting here an hour ago listening to Stephen Runo's talk, one of my colleagues' teams were having their Monday meeting. The Monday afternoon meeting is where everyone comes in with a long list of pre-selected publications of important scientific papers that will come out this week, and everyone says, well, I find this important, and I find this important, and then we discuss a little and somehow come to our conclusions of what we will actually have on the news or on the science news. If you work for longer programs, like 25 minutes, half an hour features, it's the other way around. It's really me as a journalist who comes up with an idea, thinks about how I would like to tell the story and propose it to the editor-in-chief. And then as an editor-in-chief, I decide on those proposals mainly on two grounds, one being, does it fit in the general theme of the program, and the other, is it sort of balanced out with all the other themes that I have in this, either in this show or in this week or whatever. So it's a very, us science journalists, we are all very interested in all sorts of scientific events, and what we find the most interesting and we think we can tell a story about, we propose that, and then usually that's what's getting on air. Just to give you an idea here, some of the topics that I covered in my show where many other people work for as well, but the topics that I covered myself were in the last weeks, months. The 50th anniversary of the findings of the fossils of Lucy, the Global Carbon Budget 2024, highlights of the United Nations Year of the Camelids. I don't know if you know that that was happening last year, 2024. Camels, dromedaries, llamas, funny animals. A feature of last year's Chemistry Nobel Prize for the folding prediction and the novel synthesis of proteins. A high throughput plant phenotyping platform in the biocenter. Ancient RNA, not DNA, ancient RNA analysis of museum artifacts, or coming this week, a conversation with the biologist Florian Krammer on influenza. So a lot of different subjects, and obviously I cannot be anywhere near an expert in all of them, not even one actually. So what makes a good science story then? Explaining the facts is obviously one point, and Actually, some see science journalists mainly as that, as translators of scientific results. I was lacking the word. So many think that that's our main, that should be our main occupation. We talk to you scientists and make sure that on the other end of the pipe, the layperson understand what you are saying and probably also understand why that is important. This is an important part of science journalism, I would say, but it's certainly not all. A science journalism piece should be true to the facts, of course. It should be understandable, very important. It should also be appealing and interesting. The people who listen to us do that in their spare time. None of them has to. And if we are boring, they turn off the radio, they put away the newspaper, whatever. So we have to also make sure that the way we are telling those stories is interesting. And of course, we have many tools that help us get there. What I tend to do a lot in my programs and what I find important for a more general understanding of science and seeing the fascination of science is that I tend to include a lot of making of parts into that program. So, for example, this one thing that I just said that I did in November was about the 50th anniversary of finding the fossils of Lucy, of Australopithecus. I talked to a paleoarchaeologist for that, and him, who was doing excavations in the desert of Ethiopia himself, and him telling me this. When we go there, we live in tents. There's a stone desert, and these wind devils who come along, and we dig in the heat of 45 degrees in the shades, only that there are no shades. That's a way of what you create there is a picture in the mind of your audience, and they are somewhat there with you and are then probably more willing to listen to all that maybe a little more boring stuff about how you can detect from these fossils that this creature was walking on two legs, which really is the big thing about Lucy. It's the first fossil of a hominid that walked on two legs. But what I try to do is sort of drag you there, take you there to find that interesting. I hope I could convince you now that this was not too boring. Another point that we often try to cover in our stories is the question, what for and why now? If I talk about applied sciences, that's obvious. I mean, if you're working on a certain technology, if you're working on a certain medical improvement, the question what for has an obvious answer. But even for all those curiosity-driven scientific projects, for all these other things happening in science, I consider it not only super interesting, but also our responsibility as journalists to at least try and ask that question, why is that important now? What might be a societal context why science is looking at these questions now, why certain scientists are looking at these questions? And very often, as we all know, science has not only societal, but also political, ethical, or even philosophical implications that we may want to try to cover in our stories as well. When it comes to controversial issues, there's a couple of things that we try to consider. Of course, the most obvious is we try to keep our equidistance to the different sides of that controversy. We try to get as many opinions in our program as we can. There's a big caveat to that, though, that is called false balance. How many of you have heard of that? That is something that we really fight a lot discuss a lot in our teams. False balance mainly means that if you, and climate change is a very, very good example for that. If you have a society of scientists at the IPCC and out there in the world, where 99 point I don't know what % of all those scientists say that climate change is human-made, and you have 1% who says it isn't. And you always have people coming to us and say, why don't you talk to them? Why don't you invite one of them to give you the counter arguments? And I say, well, it's a sort of, if I want to have that balanced on my podium, I'd have to, or on my program, I'd have to have 100 people there, 99 of them being the scientists saying it's human-made, and one being the one saying it's not. This is something I cannot possibly represent in a program. So if I do a program and condense it down to this 1% and one person of the 99%, I get a discussion that looks as if it were 50-50. And that is just not true to the facts. So how do we come around there? How do we still include the fact that there is people who are of a different opinion? How do we check those facts? How do we try to convey that we are not too one-sided, but do not fall into this trap of a false balance and of following every argument, no matter how stupid it is? That is really, really difficult. In another example, and with that I'd like to come to an end and back to the subject of this symposium. It's been about a year ago that there was there was something new to come out, a new discussion in the European Parliament about the regulation of the new genomic engineering techniques, and I invited Hermann Buerstmayr and Andreas Heisenberger from the Umweltbundesamt, the environmental agency Austria, into my program and had them discuss the facts of the pros and cons for how this technique is regulated. And I would say, from my point of view, that was a very fair exchange of arguments, of sound arguments, where you could also see why what I find interesting is that in these discussions you always see that people are arguing on different levels. You were the scientists talking about how this is being done, what one can do, what the advantages are, and then there was the person from the Umweltbundesamt who had a completely different mindset, and from that mindset argued in a different way. And that, I think, is interesting, and that, I think, is our responsibility as journalists also to convey to an audience and to make understandable to an audience. So let me just stop with saying, what do we journalists need from you scientists? We need information and your expertise, obviously. I don't know, I mean, some of, especially in the first row here, there are sitting some whom I've known for many years and whom I know I can call anytime and ask some background about scientific questions, new papers, whatever. That is extremely important for us, that you are there and there to give quick answers. We need you as interview partners, especially for radio. Obviously, that is sometimes not quite as easy because then you need time and we need to tape it. And we are very happy if you agree to our invitations. How many of you have ever made a media training? That I would like to know. Okay. I thought it'd be more people by now who do that, who learn all this. Of course, I don't know in detail what you did in your trainings, but everything that you might have heard, use layman's terms, use simple words, use stories out of everyday's life to make people understand the point you want to convey. All these things one can practice if one wants to, and are things that then help us as journalists if we interview you and put these interviews, for example, on radio. So it's nice if you do that. We very much appreciate that, but you don't have to do our job. If you don't want to, that's also fine. That's what we are there for in the end, really. Thank you very much for now, and if all the questions that you have, I'm happy to answer later. ausgeschaut, dass ich auf den Punkt fertig werde.
Perfect. Thank you very much, Birgit Dalheimer, to sharing with us the life of a science journalist. And I can understand that this is an exciting job. You learn a lot of different things, and you have to learn a lot of different things. Any urgent questions to Birgit Dalheimer right now? If not, we still have time now for about half an hour podium discussion. So for this, I would like to ask all presenters of today to come to the front desk, and you in the audience to think about questions. So including students, we also have some questions on maybe technical details. Also, Stephen, please come here to the front, and all the speakers, and we line up here, and we are ready to discuss questions. And I can have a look in the live stream, because we also have some questions from the live stream, and maybe we can answer a few of those as well. Maybe we start with the live stream questions, because they're already here, and you think about your questions now. Can I ask you, Ortron, that you take the microphone and maybe find out who is asking a question? And here we have our microphone. Ins Publikum kann man den schmeißen. Just check in the chat what we have here. Oops, sorry. Oh. We can leave this complicated picture. So anybody has a question in the audience, you can, okay, there is one.
First of all, I want to thank Professor Steve Runo for a wonderful presentation. And now I want to ask two questions. Are there any invasive sorghum species that are sexually compatible with cultivated sorghum and commonly grown near agricultural fields?
Did you understand?
Yeah, I got the question. You had two.
Yeah, and the second question is, does trigger resistance confer a selective advantage to invasive sorghum species? And could this lead to the evolution of trigger-resistant invasive populations that negatively impact cultivated sorghum?
Yeah, thank you. Those are two good questions. The first questions, are there compatible species of sorghum that are wild and invasive? Yes, so sorghum, like I said in the presentation, has a really wide and broad genetic diversity. So there are some invasive species of sorghum, or like shattergrass, but does Striga resistance confer selective advantage that could lead into integration with invasive species? No. Like I said, all we do for Striga resistance and for looking for alleles for Striga resistance and then using genome editing, or rather genomic technologies, we rely a lot on what exists in nature. So these are already free living and already being used germplasms of sorghum or cultivars of sorghum. What we do is to try and improve them for varieties that are produced by farmers. But really good questions.
There is a question from Adolf Marksteiner to Stephen.
Yeah, is my micro open? Yeah. Questions to Stephen Runo. What about patents, and what about, say, companies who will, as a breeder, then want to pay the farmers fees for, say, resistant breeds and so on? Is this a topic in Kenya, or would you think that's not a big problem?
Yeah, again, a good question, and this question often comes, and this is also from the debate that people are having on GM technologies, with that being one of the reasons that is given for not for not using new technologies. I agree. This is a question that needs to be addressed, and addressed very carefully and very, very thoughtfully. Obviously, Kenya and Africa is entering into these new technologies and genome editing, for example, way after many countries have done and many companies have done their bit in research and development. So they have a budget that they use for research and development. But if you ask me if the intention of those companies right now is to recoup those profits from farmers that are using those technologies, I would say no, I don't think that's their priority. But having said that, I think this needs to be evaluated very carefully, and good licensing models, if they need to be addressed, they need to be thought through very carefully and beforehand. I don't think that licensing would be an impediment, at least for genome editing for now. But a lot of people that do not advocate for technology such as this cite that as an example. But I think very aware of that and needs to be thought really, really carefully and thoroughly, and come up with good models that would not infringe on copyright, but that would allow technology to be deployed.
There is a question here. Yeah.
Thank you. Thank you on all of the podium for the interesting tellings. And I want to support Mr. Marksteiner that we need a new narrative. And especially, in my opinion, there's a big difference between GMO and new breeding technologies. I think we will have no chance that genetic technologies or something like that the consumers accept in Austria. So I have the question to Mrs. Dalheimer. Do you think it's realistic that we have a chance that the normal consumer accepts that breeding technologies, new breeding technologies, are not GMO? Do you think this narrative is possible?
Well, I guess yes. I mean, I guess a new narrative is possible in a certain way. I think a lot has changed already. I mean, when I did my master thesis in '97 at Margit Leimer's lab, that was the year of the Volksbefragung, that was the lab where the elder among us remember, where these apricot trees resistant to viruses were made. I was there as a master student when journalists from all over the place came to do reports on what we did there. It was funny. I mean, I had to wear lab coats. You probably know all these stories. We were made into those little witch kitchen things. I don't know. And I think already a lot has changed since then. I find it, for example, extremely interesting that the one program that I mentioned that I did with Hermann Buerstmayr and Andreas Heisenberger from the Umweltbundesamt last year on, call it new breeding techniques, call it new genomic editing techniques, there was hardly any reaction from the audience. 10 years ago, do the same thing and you get thrown apples, tomatoes, and raw eggs on you. Last year it was like, okay, another. So I don't think it's such a shocker anymore in a way. That's my my impression, at least from the reactions that I get from the audience. The new narrative thing, I don't know. Honestly, I don't know. I talked to Urs Nigli about it, so you said it comes from him, this new narrative thing. He's one of the, most of you probably know him here anyway. He's one who's very deep into this organic farming, but also somebody who says we might adapt genomic editing into organic farming one day. And he told me that, of course, he had lots of not very nice conversations with organic farmers after he first said that, but it shifts, it slowly shifts. I guess much more than on a new narrative, really. It depends on which NGOs run which campaigns, which politicians say what on the subject. I guess that's much more important than finding a new narrative.
Maybe I can start here with a few questions from the chat group who is following us on YouTube. Maybe an important one that goes exactly in this direction is a very direct one. It says, do you also include ethical and ecological aspects in your reframing of the NGD narrative? So this is exactly one of the questions. Maybe it goes directly to me because I designed the program today. Of course, in one afternoon, my short answer would be in one afternoon, we cannot include all aspects. So definitely, we have not invited today environmental scientists or ecologic experts in ecology here for this one event. But I know it has to be discussed also from this perspective. And ethical aspects, I'm not really sure what is meant with it. But I think we can look at ethical aspects from different points of view. I think it's a very ethical project, what Stephen Runo is doing, because this is a project which has in mind to help small subsistence farmers to cope with a big problem they have. So I would say the target that you use or that the aims that you have is the ethical question and not the technology itself. So my most easy explanation on this is a personal one. A few years ago, I had a mountain accident, and I broke my leg. And then, luckily, we could reach the rescue service, and a helicopter was coming and saving me. So for me, and brought me to the hospital, and I was, now I can walk with my 2 legs again. So for me, a helicopter is a super good technology. I'm so happy that we have helicopters that brought me to the hospital. If I would live in Syria, I would think a helicopter is a really dangerous equipment. Because a helicopter in Syria, if you were living in Homs or some other cities, typically dropped bombs on you and did not come to rescue you. So the helicopter can be a nice thing for me, can be a very nasty thing for other people. But what you do with your technology and how you use it, I think this makes most of the difference. And this is, in my point of view at least, how we should address the ethical questions. Do we use the technology for the right things? This, I try to answer, hopefully, our audience. This is my point of view. But you can write me back and also can respond by email. But I have 2 more questions that go specifically to Stephen. Thank you for the presentations. Are striga-resistant plants already on the market? Have farmers access to it? And what is the experience with these new types on the market? And the second one is a very similar direction. Most smallholder farmers rely on farmers' seed systems and the informal seed sector. How to conserve their sovereignty and make them dependent? Also, the question is, will these new seeds make farmers more dependent on outside sources, and is this a bad thing? Or maybe a general question, how is the seed system for smallholder farmers at the moment, and how would it be impacted by the new types of varieties that your institute is also working on?
Yeah, good questions. So the first question, I don't know if the person that asked that question means edited varieties. No, those are not in the market. But we do have striga-resistant material that we identified. So this would be germplasm that is grown elsewhere, so maybe grown in some parts of Ghana, some parts of South Africa, but not known to have striga resistance. Nobody has checked if it has striga resistance. And so what we have done is to collect all those germplasm, used a high throughput method to detect whether they have their alleles that make for Striga resistance. And the good thing with that is that you can already test those in fields, and you can already advocate them if they are not registered as varieties that farmers can use to manage Striga. Then you can initiate the registration process. And if they're already been used by farmers but not used for Striga resistance, then you can advocate them for Striga resistance. And those are already in the market. Some of them are already in the market, and farmers can access them from agrovet stores or from other seed companies. And then the other question is a really, really good question about seed systems. And I know and I agree that our seed systems are not very optimal. Farmers tend to use seeds and then exchange them, save them for this season, and then and share seeds. So that needs to be worked on, and we're working on how to best develop the seed system so that the integrity of the trait is maintained. We do have right now the Kenya Agricultural Research Institute having their own seed company. We do have talks and discussions with farmers, telling them on the importance of maintaining seed integrity and telling them that this will only give you the advantage of high yield if you get if you use the right seed. Not very optimal and really good contribution, but something that we have in mind and something that we must do to make sure that our seed systems are robust enough and to make sure that the seed systems allow for integrity of the trait and make sure that farmers are aware of this and the advantages of maintaining this.
And is there a fear that farmers get, like we sometimes hear in our discussions, if we use high technology, then it's only, how to say, in the hands of a few big multinational companies, and then we get dependent on them. This is one of the discussions that we often hear. Could this be a cause of concern also for your seed system?
No, I don't think so. And we don't have that, to be honest. That was a narrative that kept getting pushed. Technology victim mentality, which we also have to go away from. But it's not an argument that it has been fronted, but it's not something that that farmers have as a perception in Kenya. No.
Okay, thank you. So that was the question. Yep, please.
Yeah, thank you. Is it working? Yeah. Thanks to all of you for providing this very interesting afternoon. I have two questions to Birgit Dalheimer, to your talk. I found it quite interesting that you said that NGD topics are not a shocker anymore for the Ö1 audience. And I wonder, like, as we also experienced, we are highly biased and we are living in kind of a bubble. What kind of methods or strategies do you think would be applicable to reach, like, outside the bubble, people that are really cons and have like very solid opinions on these topics? And my second question would be, how do you decide what's interesting topic-wise? Do you somehow react on happenings or news to decide the topics on? Yeah, thank you.
Okay, the first question, I was also as a short reply to what you just said, if I had been here as a journalist to cover this afternoon for a program, that I would have had to go out and talk to somebody who is not in this bubble. I had this, I'd have this feeling that I have to collect the other arguments as well. I say something that's probably completely naive. I mean, I know it's naive. I'm working for Ö1, and I know that I have long programs. I can take 25 minutes. You might find that a short time, but for news items, that's a really long time, and I can tell the arguments, and it's almost pathetic to say that, but I believe in the Enlightenment. You know, I can give people the arguments and they can think about it. I believe in is too much, but that's what drives me really. That I try and find what everybody is saying for and against the use of these technologies, and if the argument as such is sound. I mean, I don't go for false facts or alternative facts or what they are called nowadays. But if the argument is sound, I will put it in. I will include it in my programs, and then people out there can make up their minds themselves. That's what I also meant at the beginning of my talk by I don't see my role as being the one who has to increase acceptance for anything that science does. I convey the information, I try to make it interesting, I try to make it understandable, and I try to show where the arguments come from. But I trust my audience in controversial issues to make up their minds themselves. And I have the luck to work for a medium that allows me to do so. You might add a long discussion here on journalism and the role of journalism and the means of journalism and the means of science journalism there's one newspaper in Austria that I know that has a really big science department, but everywhere else it's freelancers, you've heard all the arguments. So I cannot say more than that because I'm already on the safe side working for Ö1, so to speak. And sorry, now I forgot your second question. How we decide what's interesting. Of course, we go along the lines of, especially in the aktuelle Wissenschaft, so the daily business Wissenschaft. Monday morning, I get press releases from at least 15 different weekly science papers. So Nature, Science, Cell, all of them. And they already make a pre-selection of what they think is super interesting in their current issues. From that list, I take the ones that I, Birgit Dalheimer, personally find interesting. One. Then I take the ones where Austrian institutions or Austrian scientists are involved. That's also always, we report on that rather than from anywhere else. Then I talk to my colleagues, and very often, funnily enough, we all find the same things interesting. So maybe there's a certain feel already. Then we get a calendar from the press agencies. Austrian press agency has a calendar out every Monday saying what's on press briefings, press conferences, new studies coming out in Austria this week. That is something that goes into these weekly meetings as well. And then we say, this is the list of interesting things. This is the list of people that we have. This is the list of program that we can fill. And then we take a pick, more or less.
Yeah, just
Sorry.
follow. Well, go ahead,
Yeah, so I think it's not a question, but maybe to congratulate Claudia and Irma. Really, really good work. Very impressive. And maybe just find out from them what they think about this, because the young generation now is very savvy with computers and very savvy with programming. Do you have a place for AI? Because I know AlphaFold as well as Vina and docking technology can probably help you refine your targets. Have you thought about that? Then also have a question for Herbert and maybe a suggestion on and this is something that I picked during the discussion and also listening to D. Marksteiner about having a different narrative on, I think something that piqued my mind was the fact that we all eat cereals, maybe here more than us, and that if you consume a certain amount of cereal, a certain threshold, then you have the risk or long-term effects of having poisoning from mycotoxins. And the fact that you're developing and the fact that you have an ability and a technology that can help reduce mycotoxins, is that something that can be used as a SAP or the new selling point? Just thoughts. Anybody can answer, whichever. Maybe Claudia.
Yeah. Well, so
yeah, I think that obviously nowadays the bioinformatics field is gaining more and more importance, and actually, yeah, like I think there's at least like one basics bioinformatics knowledge is needed in order to process all this data that we are generating. So, well, I don't know, my colleague Irma has something else to add. Me personally, I would like to have more knowledge on this bioinformatics field, because I think it's a very useful field to indeed arrive to conclusions from experimental data, to process this data, and to arrive in a time-effective manner to other conclusions that maybe some years ago was not possible.
Mm-hmm.
Yeah, I agree with Claudia completely. I also would like to add on different versions of CRISPR-Cas that are used now, different forms of Cas9 nuclease, different. So you can tailor your project using different CRISPR-Cas versions, and that also can have an impact on how people and how politics perceive your project, because some of the modifications made by CRISPR-Cas, they are actually not considered to be GMO, for example, in USA. So I would say that this development of the technologies really open up all the doors for projects such as mine and Claudia to be incorporated into society and being accepted.
Yeah, I will take up the question concerning the AlphaFold, and you mentioned molecular dynamics, because I'm not actually a plant breeder, I'm more like a biochemist, and so I work with enzymes and I also do protein structures, and I mean in experiments, so we actually still do crystallography, and we are discussing a lot this AlphaFold, and I mean, it's a super cool tool and really, really helpful, but in many cases it doesn't answer our questions, because when we talk about protein structure, AlphaFold can give us a good look how this thing overall looks, but the details we're asking for we mostly get only from experiment. And there is molecular dynamics. It's also interesting, but it's not that simple. So molecular dynamics is a tool when you ask the right questions. So when you just simulate something, you will not get a straight answer. When you have a detailed question, you might, because this is really computational complex, and you need to narrow down your questions so that you can calculate it in a lifetime. So basically, I think these tools, even bioinformatics, is really helpful, but probably I'm old school, and I think that good old experiment is still necessary.
Okay, I think we had a few more questions. Yeah.
I do have a quick question, yes. So thanks for the presentations. I'm a bit hidden behind this column here, so you can go to the left and to the right. The question is, it goes to Stephen. If I look at the picture over you there, you're sitting like in a field, and my question is there a lot of paperwork necessary for doing field testing of these new sorts? Or how does it look in Kenya, for example?
So paperwork for doing field testing, so if you're asking on the germplasm, if I'm testing germplasm for field evaluation for Striga resistance, of course there is ethical approval and all that accompanies that. Also depends on if it's a GM, if it's not a GM, and if it's edited. So there are different categories and paperwork for each one of these. Are there protocols that you need to observe when you're testing for, and ethical considerations when you're doing field testing for Striga resistance in a farmer's field? Yes. There are protocols and procedures that you need to observe if you're doing and working with Striga, whether from one field or from the next field, whether from a farmer's field or in the lab, there are protocols and there are procedures that you need to observe. Obviously, you don't want to be transporting striga from one field to another. You need to observe sanitation. You need, if you're using striga in the lab, it has to be cleared. You have to show that you're able to destroy everything that, so that you don't have striga lurking around. And if they found striga in Nairobi, it would be most likely be me. So we take all precaution over and above the procedures that are recommended.
So I have a technical and a legal question. So first, I didn't really get, how do you transform sorghum? Are they stable transformers? And before you go to the field, do you have to outcross the GMO part? And so what are the regulations that you can go to the field? So here at BOKU, as long as we stay in growth chambers, we can more or less do what we want. But obviously, you can produce wheat and barley and whatever much faster than you will acquire growth chambers and rooms to test them. So this is, I think, a real bottleneck for science that you obviously don't have. No? So lucky you.
Yeah, good question. So how do we transform sorghum? We use agrobacterium-mediated transformation. We use protocols for basic protocols for transformation. Agrobacterium-mediated transformation is the one that works best for us. And I should say this, it's not everybody that is allowed to do genetic transformation. If you're doing genetic transformation for sorghum, for maize, for anything else, it has to be done in a biosafety level 2 lab. In Kenya, there are only 3 such labs. Ours is one of them. And the biosafety lab has to be inspected and approved by the government, Kenya Plant Health Inspectorate Services, as well as the National Biosafety Authority. So the very act of doing genetic transformation, whether you're doing it for gene editing, whether you're doing it for sorghum, for commercialization, or whether you're doing it for just experiment, it has to be in a certified lab. And that needs to be inspected, and you need a permit to be able to do that. So that's one level. If you want to do genetic transfer, if you want to do a field study, like we have done before for drought in maize, like we have done before or that's ongoing right now for cassava, not by our group, you'll need to apply for a permit, again from the National Biosafety Authority, for what is called a confined field trial. So you prepare an application that goes to the National Biosafety Authority. It is evaluated on its merits, and you have to know, you have to check every box that says you adhere to environmental safety, you adhere to use and transportation of the genetically modified organism. If you want to do a gene-edited experiment, for example, so you have done, imparted the gene, for example, if it's, say, if you have done a striga-resistant gene editing experiment, and you have segregated out the gene that, or any genetic material that would be termed foreign. So if you have Cas9 that you used in the genetic transformation, but you did it in a way that you can segregate those out according to Mendelian genetics, then you show proof to the National Biosafety Authority. So you apply to them and tell them, I have the sorghum variety or the sorghum mutants that I've developed using editing, but I have removed any foreign genetic material, and this is the process that I used for doing the segregation. So you apply, and within 90 days, the National Biosafety Authority will come back to you with an answer and tell you we are convinced that you don't have any genetic material, foreign genetic material, in which case you are okay to proceed, not by regulation, under the Biosafety Act, which regulates conventional genetically modified organism, which would require you to do a confined field trial. So if you show proof of that and they are okay with it, then you can proceed and do your field evaluations with restrictions, without restrictions of a GM. If they have questions, they come back to you and ask you, we are not clear about how you did this, we still think you have this foreign genetic material, and then you answer back, and if that back and forth and iteration, they're convinced, then you're free to do the field evaluations. If they're not happy, then you still have to be regulated under the Biosafety Act.
Okay, so I think we have 1 or 2 more questions. Yeah, Judith?
Yes, thank you very much. I have a question for Stephen. There's a previous question where you had a chance to touch a bit on the seed system in sub-Saharan Africa, where like a large percent of it is really contributed by the breeding companies outside of sub-Saharan Africa, and they produce the seeds and then multiply in sub-Saharan Africa, and then later on the seed that is sold to the smallholder farmers is kind of expensive according to the smallholder farmers. So my question goes to like, how do you see in the long run such kind of technology to be employed in this breeding system? And even if you have a breeding company that originates in Africa, do you think you'll be able to produce seeds that are easily accessible in terms of price? Because as a smallholder farmer, you have just a small land, and you're looking forward to have, of course, income. And if the seed is expensive, you can't really get what you're doing, like the investment will come back. And if you have a hybrid seed in the market, which is expensive, you are going to produce a new technology seed. That how are you going to compensate and also convince the farmer, use this seed instead of the seeds that are available? So it's either we go back to farm safe seed or what. So that's my real question. In the long run, how is it going to be?
Yeah, very, very good question. And I think we need, the bottom line is we need to develop our seed systems. There are many, many startups that are coming, local startups, local startup seed companies. In fact, I know one of them, a colleague that is starting their own seed company, and these are the ones that we need to promote. I think local seed companies would work better, and if they are empowered in terms of supporting those startups is something that we are hoping to do, because really not optimal, not ideal, I agree, but we need to move and see what direction to take with the seed companies, with the seed systems. I should also say that one of the most successful seed companies in Africa is actually African. So Seedco, which is based in Zimbabwe, has a very wide market niche in Kenya, and farmers actually like getting seeds from Seedco. So, promote local startups, and I think this would make things cheaper and more accessible. And we are doing that. And not to say that seed companies or seeds that come from multinational companies are bad, but I think we should also promote our own startups and give them an opportunity.
Yeah, this is a very important point. The best technology is pretty useless if it doesn't reach the people who need it. Yeah? So there needs to be a lot around it to make it really useful. I think, Otron, you have a question?
Also to the young scientists. So you get this excellent training here. You acquire all this expertise in handling the technology. You have interesting topics, targets, whatsoever. But in the end, you will face the situation that your work is not appreciated within Europe. So to make it clear, and you can only answer with yes, would you consider to move your future workplace to the countries where you can see that your work will be in the benefit of the farmers somewhere. So are you ready to move to Canada, Argentina, Kenya, where your expertise is more appreciated than here? And this comes with a question, is Austria and Europe clever to risk such a brain drain to countries which do not profit from the investment they have done into your education?
Yes, I got more. and no, I don't think the European Union is clever by putting such a ban on or having such an idea. But I do believe that maybe not in the recent years, like in the recent future, the very near future, but I think that in the end, and I hope that maybe our new generations would have like much more scientific knowledge and could be more open to change the situation that faces Europe right now.
For the optimism.
Okay, I think, yeah, and this is also an experience I have. I was in a big meeting with lots of students talking about ecological aspects of agriculture and reducing pesticide use. It was 300 or 400 students in the room, and in the audience nobody talked about new technologies, but the questions came from the students. We want to reduce pesticide use. Why don't we use the latest technology to make the plants more resistant? And then some people those were my hair color, started to freeze. What should I say now? Because they are challenged by the students with exactly these questions. But I think we have to come to a close. Maybe I have one more, maybe final sentence that also fits in our program from the chat of our followers online. I imagine acceptance will increase if the labeling of products remains transparent, consumers and breeders have the choice, risk assessments are being taken serious, and the products keep their promise. And I think this is maybe another point of consideration. Thank you, all the people also online and in the chat to ask your questions. Sorry we could not answer all the questions, particularly we did not answer now the technical questions, but I ask you out there at your TV, send your question to the speakers. If you have a technical questions on how you do the transformation, how this and how this, I'm sure all the speakers here are ready to answer your questions, or you can approach me and I can forward the questions to the respective person. So thank you very much for joining us online. Thank you very much all of you here for joining us today.